Good afternoon, everybody, ladies and gentlemen, dear colleagues. Excuse us for the late beginning of the sessions, but we are expecting our president of the university to join us. We will begin with the presentation of Dr. Ellie Barnes. Dr. Ellie Barnes' presentation is about hepatitis C virus, hepatitis B virus vaccines, and hepatocellular carcinoma therapeutic vaccines. Professor Ellie is a professor of hepatology, and she is hepatology and experimental medicine. Professor Barnes leads an established research group with a focus on applied immunology relevant to liver diseases, including cancer. And she seeks to translate laboratory and clinical findings through to human experimental medicine studies. Professor Barnes' group is developing T-cell pangenomic genotypic vaccines for hepatitis C virus prevention and hepatitis B virus cure using simian, adenoviral, and other viral vectors. She is also assessing COVID vaccines in disease cohort, and she also you work in another place. And
yeah, I've also got a program in early liver cancer detection, which is very relevant. In Egypt at the moment. Yeah, thank you.
Let's begin. Thank you.
Okay. Right. Thank you so much. It's a huge honor to be invited to Cairo. We've had a fantastic morning being taken around the first-class research facilities, Ain Shams Clinical Research Center. So thank you for the morning and for all the excellent facilities, and the demonstration of that. It was really interesting for us to see that. So, I'm going to talk to you about vaccines for hepatitis C virus, the hope and the challenges. You will all know about the rollout program for DAAs in Egypt. It's been a kind of international success that you can all celebrate. But I'm going to talk to you about vaccines today, why we don't have one yet, and why I think that there's hope for one for the future. Right, I'm going to start with a little bit of history. I always promise that as I get older, I wouldn't talk about the history of the disease I work on, because it would make me seem really old, but it's hard to come to Cairo and see the pyramids without kind of mentioning age. I mean, your pyramids are really old, but hepatitis C is way, way older. And, humans have lived with hepatitis C for probably, it's estimated on the latest publications that have come out that the genotypes evolved, the separate genotypes, 100,000 years ago. Your pyramids are here on my scale here at about 5,000 years ago. And actually 20,000 years ago is the origin of the coronaviruses for SARS-CoV-2. So Hep C is way older than all of this, and it's exquisitely evolved to exist and cause chronic disease in humans. That's why we have asymptomatic infection for so long before people get end-stage liver disease, cirrhosis, and liver cancer. And hepatitis C is exquisitely evolved to exploit the liver niche. For those of you that don't know much about the liver, let me tell you, it is the most beautiful organ in the body. It has this huge range of physiological functions, and one of those is immune tolerance, to dampen down the immune system, because it's constantly exposed to antigens from the gut. And that's why hepatitis C, hepatitis B, and indeed malaria that we'll hear about later, have all exploited the liver as parts of their life cycle, or even the main life cycle. Okay. So I think we have lived through a really incredible time over the last kind of 10 years of hepatitis C virus research. Just to put this really quickly into historical context, we knew hepatitis existed in the 17th to 19th century. There were kind of descriptions of soldiers getting jaundice after they went to war. And in the 1930s, it first became apparent that when you gave people yellow fever vaccine, they had hepatitis after you gave them this vaccine. That wasn't because they were reacting in a negative way to the yellow fever vaccine, it was because it was contaminated with hepatitis C virus. But not until 1938 was the link between hepatitis and infectious serum first made. In 1965, hepatitis B was discovered. 1973, hepatitis A was discovered. But even after 1973, it was clear that there was still this pathogen that caused non-A, non-B hepatitis, that this could be transfused between patients and indeed to chimpanzees. And it was an envelope virus. And we knew it was envelope because you could filter it and inactivate it and remove infectivity from the serum. So we knew there was this virus, but didn't really know what it was. And then in 1989, Michael Houghton, along with others, identified the hepatitis C virus. So this was the very first time any pathogen had been identified using molecular technologies. So that's the first real breakthrough for this virus. And it was identified then as being an RNA virus, 10,000 nucleotides in length. After that, it took 10 years to develop a cell culture system. And this is also an issue for vaccines at the moment. We still don't have a perfect cell culture system for hepatitis C virus infection. There's no small animal model for hepatitis C, so you can't test the vaccine in animals. You can test it in chimpanzees, but animal experiments on chimpanzees are now outlawed. There are no cell types that are permissive to hepatitis C in the lab, other than liver cancer cell lines, which have their innate immune system switched off to allow that to happen. But eventually, we did get subgenomic replicons and eventually full virus replication in liver cancer cells of just 2 specific strains that had caused fulminant hepatitis in a patient. And that allowed this golden era of hepatitis C virus research to follow, looking at virus life cycles, virus-host interactions, and it was really that platform that led to the discovery, the ability to test the new drugs and the DAAs. I would argue that after SARS-CoV-2, HCV is the most studied RNA virus in the world. That led to this therapeutic pipeline in 2014. There are 54 molecules on this dot plot chart here. So this is just a description, really, of all those molecules going from phase 1, phase 2, phase 3, phase 4, that ultimately led to these DAA combinations that we now use in our patients, a mixture of NS5A inhibitors, protease inhibitors, and polymerase inhibitors. So my question really for you is, if we have these fantastic drugs, which we do, why do we need a vaccine for hepatitis C virus infection? And I'd like to just tell you why. There are still 56 million people in the world that are thought to be positive for hepatitis C virus infection. Viral hepatitis kills more people per year, so that's hepatitis B and C, than malaria or HIV combined. There's 1.7 million new infections per year. The epidemic in some countries is increasing, in particular the United States, where there's an opioid epidemic at the moment, and they are increasing funding into hepatitis C vaccines in the States. And this is a nice paper. I've just got 2 figures here from a paper that was published very recently, 2022, and it basically describes the impact we've had on the global epidemic so far with the DAAs. And you can see that although we've treated 6.8 million people, the kind of delta decrease is really small if you look at the total numbers infected. But you'll be very pleased to see on this countrywide plot here, Egypt is a real standout country. You see that sweeping green line down? That's Egypt. You've done better in eradication than anywhere else in the world. Right, but there's still issues with these drugs. They're expensive. You need to give them for 8 to 12 weeks. They're associated with drug resistance. There are some rare genotype 4 subtypes specifically associated with triple therapy resistance. They don't prevent reinfection. Can you hear me? They can hear me.
For the record.
Okay, for the record. Yeah, that's fine. And worst of all, most people are unaware that they are infected, so they present with liver cirrhosis, or worse, with liver cancer. And furthermore, after you treat patients, as many people here know, the risk for liver cancer does not go away completely. The bottom line, to my mind, I mean, I know we're in a vaccine session, but prevention and a vaccine would be much better medicine than waiting until people get chronic infection, cirrhosis, and liver cancer. So this is just to show, really, that this is a publication which essentially highlights the ongoing risk in patients with hepatitis C after hepatitis C virus cure, estimated to be about 4% per year. So it's a significant risk even after you've got rid of the virus. And in the UK, we've very recently established a program for hepatocellular early cancer detection, because if you can identify HCC very early, you can ablate it or resect it. Most patients present at very late stage, and to my mind, ultrasound and alpha-fetoprotein is a poor modality for HCC screening. So we've established a big prospective trial across the UK, aiming to recruit 33,000 patients. We will assess those every year for the next 5 years, collect blood samples, then go back to those and evaluate those blood samples for biomarkers in the 100 to 200 patients that we predict will get liver cancer over that kind of time period. And that would be a study that you could definitely do here much better than I could do in the UK. It's called the DELIVER program, and it's funded by Cancer Research UK. Right. So why don't we have a vaccine for hepatitis C, having argued that we definitely need one? We should be able to make a vaccine for hepatitis C. Unlike HIV, for example, 20% of people who spontaneously resolve infection without any medication. And my PhD and my work after that was spent trying to understand the adaptive and the innate immune differences in those people who develop persistent infection and compare that to those who had resolved infection. And we came to understand as a kind of global community that your T-cell immune response, your B-cell immune response, and your interferon lambda 4 gene were all critically important in driving resolution. So the idea is, if you can mimic the immune response that you're seeing in spontaneous resolution, you should be able to make a vaccine for hepatitis C. So this, as I've already pointed out, is the effort that went into identifying drugs. Let me map vaccines onto the same plot. These are the effort that's been made in developing vaccines for hepatitis C. There's only 2 vaccines that have reached human efficacy studies so far. One is the AD-MVA viral vector that I've been working on, and the other is an E1, E2 protein vaccine that Michael Houghton, who discovered Hep C in the first place, has been working on. So you can see, compared to the drugs, we've barely tried to make a vaccine for hepatitis C. That's one of the reasons why we don't have one. So the rationale for a T-cell vaccine is, to start with, making an antibody-based vaccine is thought to be difficult because most of the antibodies generated in response to hepatitis C infection target the hypervariable regions, a bit like influenza in the glycoprotein region. Whereas we know from HLA association studies, chimpanzee CD4 and CD8 T-cell blocking experiments that were done in the past, and the association of the breadth and the magnitude of T-cell responses with viral eradication, that T-cells are intrinsically involved in the spontaneous resolution of hepatitis C. So we set about to try and make our first T-cell vaccine for hepatitis C. We used an adenoviral vector. We inserted into that 1,985 amino acids from a genotype 1b strain, and we gave that to students mostly in Oxford. And we started off giving 2 heterologous adenoviral vectors. What we found there was we had a really strong T-cell response to the priming vaccine, but an attenuated response to the boosting vaccine. So we replaced that second boosting vaccine with an MVA, a modified Vaccinia ankara vaccine, encoding exactly the same immunogen. And this was basically work that was coming out of the Jenner Institute at the time in malaria, which clearly showed that AD-MVA was a really potent prime boost regimen to induce a really high T-cell response. And we went on to show with this as a boosting agent, we could have these fantastically high T-cell responses, 4,000 spot-forming units on average in an ELISpot assay, which was much higher than we even observed in those patients who spontaneously resolved infection. And we did lots of work to show that this was a broad response, it was highly functional, the T cells looked fantastic, and we thought this vaccine might work. So that vaccine went on to efficacy testing in intravenous drug-using cohort in the States. 540 injecting drug users were enrolled. Half of them got placebo, half of them got the vaccine that I've just talked to you about. The results were reported in the New England Journal of Medicine in 2020. It cost probably $20 million and took more than 6 years to test that single vaccine. And at the end of all of that, this was the results. 14 patients became chronically infected in the placebo group, and 14 patients became chronically infected in the vaccination group, which was obviously a disappointment. Right, why did this potent T-cell viral vector vaccine fail to protect against chronic infection? And this is what we've been working on at the moment. And I think there's 4 possibilities that I've listed here. Pre-existing antibodies to the viral vector, HCV genetic variation, vaccine not being immunogenic in people who inject drugs, or your T cells alone are unable to control HCV viremia. So I'm just going to take those one by one. Let's start with HCV genetic variation. Hepatitis C is 10 times more genetically diverse than hepatitis B or HIV. So this is a huge challenge. And if you take T cells from the people that you vaccinated with your genotype 1b vaccine, and you test them against antigens from the different genotypes, you see a significant decrease in your T cell responsiveness. And most of the patients who became infected in the States phase 2 efficacy trial were infected with genotype 1A and 3A, and this is a genotype 1B vaccine. So that's certainly one possibility. So the way that we've been tackling that now is to make a conserved T-cell vaccine, taking the conserved regions from all the different genotypes, stitching those together, and encoding those in viral vectors. And we've taken that through to mouse studies so far, and in that way we've been able to show that we can generate T-cell responses to multiple genotypes taking this approach. So the next hypothesis, was this vaccine equally immunogenic in people who inject drugs? I mean, I had no reason to believe it would not be, but actually it's 2 to 3 times less immunogenic in the injecting drug cohort compared to the healthy volunteers we tested in Oxford. The reason for that is completely not understood. Furthermore, in Oxford, 100% of our healthy volunteers responded to this vaccine with fantastic T-cell responses, and in the injecting drug cohort, 22% made no response at all. So this is definitely an issue, and moving forward, when we're testing new vaccines in these populations, we need to take this into account. However, the magnitude of HCV viremia in that efficacy trial was significantly different in those patients who received vaccine compared to those who received placebo. The vaccine was doing something. It was lowering the peak viremia, but it wasn't sufficient to prevent chronic infection. Right, so very recently now, working with Heidi Drummer in Australia, we've been taking viral vectors and in those encoding our conserved genotype non-structural protein alongside the envelope region with the hypervariable regions deleted, aiming to make the first vaccine that generates both T cells and antibodies. And we published that in Hepatology in March of last year. So there are new approaches now, making new immunogens where we're aiming to generate both T cells and antibodies. I think this is going to be a hugely rich area over the next 2 to 5 years. People are trying a range of different things now for hepatitis C, and in a way the failure of our first T-cell vaccine has kind of revitalized the field. People needed to know if that vaccine was going to work before there was a real push to try anything else. So we've got E1, E2 protein vaccines that Michael Houghton is working on still. We've got Heidi Drummer, who's now aiming to make mRNA vaccines with an E2 protein with the hypervariable regions deleted. We've got a Copenhagen group taking a killed whole virus approach. And then there's other groups identifying potent neutralizing antibodies and exploiting the advances in structural and computational biology to put those on HCV glycoproteins, on nanoparticles and viral-like proteins. And we've been under very recent discussions with Ahmed about whether we could think about making an R21-like vaccine, which we're going to hear about from Adrian in just a minute, with the HCV glycoproteins in that instead of malaria. And then vaccines that aim to induce T cells include peptide vaccines and epitope strings. I think those are a thing of the past. Viral vectors to deliver large immunogens, and mRNA vaccines, of course, definitely are going to have a role to play and need to be evaluated. And there's recent efforts to bring together all of these approaches. So I would like to end there, and I'd like to say that the discovery and the cure of hepatitis C, to my mind, represents the best of modern medicine. Novel approaches are needed to make new hepatitis C vaccines, and as I've already highlighted, that's happening now, but this is much more challenging than we saw for COVID. One thing I haven't touched on is how we're going to test these new vaccine candidates as they come out. This is a huge issue. It took, as I already said, $20 million in 6 years to test one vaccine. That's not something which we can repeat with multiple vaccine candidates. So one thing the field is talking about is whether we can set up human challenge models for hepatitis C, like we've done for malaria and salmonella and other pathogens. And I think the drugs are so effective, you could now have a human challenge model for hepatitis C that would allow you to benchmark one vaccine candidate against another before you went through to phase 3 or phase 2 testing. And lastly, I'd like to say that new technologies for liver cancer detection and risk prediction, in addition to hepatitis C vaccine, in my view, is urgently needed, and it's definitely needed in Egypt. So I'd like to end there and thank everybody for listening to my talk.
Thank you, Professor Barnes, for your very interesting presentation, and we hope you the best of luck to finish your researches and the clinical trials and to appear in the market with this new vaccine just to end the hepatitis virus in the world. Before you leave, is there any questions for Professor Barnes?
Sorry, I'm happy to take questions. I thought they were at the end.
Yes, Dr. Professor Abdurrahman, please, the mic.
Thank you for your very nice presentation. For me, hepatitis C virus is just an RNA. It's just an information that got embedded inside the hepatocytes. So for me, the problem is from the time of get exposure to the time of the virus reaches the hepatocyte from inside. Once it got in, it's almost game over for vaccine to work because it's embedded now in my DNA. As you mentioned in your data, the treatment sometimes don't prevent the re-exposure and don't prevent the virus from being reactivated again in my liver. So my question is, do you think that the role of neutralizing antibody will be the most efficient way to prevent or to be used for vaccination, to prevent the virus from going from the blood to the hepatocyte, to be neutralized during his journey from exposure to infection?
Yeah, thank you. That's a very good question. I mean, I'd just like to challenge you a little bit on the point that hepatitis C is kind of in your DNA. It's not in your DNA. It's not like hepatitis B, which is in your DNA. It goes through a life cycle in the cytoplasm. And mostly after you've cured a patient, even if you immunosuppress that patient, it does not reactivate. I mean, I know there are some case reports, but it's not like hepatitis B where actually that's a very common clinical scenario.
But I mean, once it got inside my hepatocyte start to replicate, the role of vaccine will be gone. Well, that's why I'm focusing on the role of neutralizing antibody.
Okay, sure. So it would be much better if you can prevent that from happening, and you would need a panel of potent neutralizing antibodies to prevent the virus from getting into your liver. Nevertheless, natural history studies tell us that patients do become infected. They become jaundiced. The virus has definitely gone into their liver to the point where it's caused a hepatitis. And those are the patients that actually clear the virus. And they become cured after that naturally with their own immune response. So it's definitely not game over for that 20%. So it's possible, because natural history tells us it's possible, and it's your T-cell response that's doing that.
I agree with you, but sometimes after 6 months of getting the treatment, sometimes we find some cases that produces recurrence of the infection again during a very short time period. I don't think this is because of another exposure to virus. I think it's because the virus is already deactivated inside my cell somehow. Maybe not aware of the whole mechanism, but to have a recurrence infection just a few months.
But that's very unusual. I mean, it happens, but that's not the common clinical picture. The common clinical picture is you give 8 weeks of treatment and people are cured for life. That's the normal clinical picture. But you know, I agree this happens, and it will be a challenge for a vaccine.
Yep, thank you. Thank you for your presentation.
Thank you. Is there any question? Yes, please.
If we succeeded to have or obtain a vaccine for HCV, how long would it take for us to develop a new vaccine? I'm asking about the viral genetic variability or mutations. Is the virus stable enough for us to have a vaccine that would stand for many years, or would take decades, or what would be the time frame for that?
Yeah.
I don't have an answer for that. We don't have research on that.
No, and I don't have an answer to that either until we give it a go.
Any speculations?
There are. I mean, all I can say is there are people, especially injecting drug users, who have been repeatedly exposed to different genotypes and who repeatedly clear. There are also people that become reinfected, but there are definitely many people who repeatedly clear injecting drug users. I mean, Andrea Cox has published some very nice data on the Bashi cohort showing this. So I think it is possible to generate a protective immune response.
So I'm
against
afraid
multiple
if we
genotypes.
develop a vaccine, it will be valid for months or something, or it would be valid for more than years. That's the question.
Yeah.
Because if it's for months.
I don't think hepatitis C, once you've got chronic infection, evolves like influenza or even COVID at those kind of rates. It's not as rapid as that. I mean, I know we have these distinct genotypes, but those are generally fairly geographically confined, you know. Yeah.
Much appreciated.
Thanks.
Thanks.
Thank you. Thank you for your question.
Thank you. Is there any other question, please? Yes.
Thank you for coming, and thank you for an amazing presentation. My question is based on a previous proposal I made 11 years ago under supervision of Professor Dr. Sahar Shulman. My research was focused on the virus's entry and absorption stages. And I aim to produce a synthetic protein vaccine for HCV. These synthetic proteins resemble the viral envelope proteins E1 and E2. I aim to not only to produce antibodies for the virus, but also to block the receptor sites without their entry inside the hepatocytes and without the immune system response to these proteins. I know that there are a lot of factors that help in the virus entry inside the hepatocytes. Despite these factors, can we find a way to achieve and apply this strategy or not?
So, yeah, can you just say your last sentence
like
again?
That, to produce a synthetic protein without entering the hepatocyte, but also produce antibody for the virus.
Yes. I'd have funded your grant if you were writing a grant to make an E1, E2 protein vaccine. I think there's not been enough done in this area. I think the entry factors for hepatitis C are multiple and complicated. And I don't know what the redundancy is in the system. I don't know if you were to block one or two of those viral entry receptors, the virus could still get in with the other three that are known to participate. But I would have thought blocking any of them may be effective. So I think these are really good ideas.
Thank you.
Thank you. Can we go to the next speaker?
One question.
Thank you. Oh, I'm so sorry. Okay, another question from Dr. Khaled. No, no. Please, the mic here, please.
First of all, thank you so much for this nice presentation and comprehensive presentation. Actually, as we know that developing a vaccine against HCV is globally very important and challenging at the same time, particularly in Egypt, since we are endemic. Egypt is endemic in HCV. Yeah. You have mentioned that you have reached two potential vaccines, one enter clinical phase 1 and the other one enter phase 2 vaccine. However, both were failed to clear infection from, or to avoid reinfection in chronic patients. My question is, the T-cell vaccine and B-cell vaccine, have you tried to combine two or more vaccine candidates for such? Have you done some or certain studies for combining these two types of vaccine?
Yeah.
This is one question.
Okay,
the second one is about the vaccines that you have tested and enter phase 2. All these vaccines have been tested for naive persons or for chronic individuals? Because as you know, the chronic patients suffering from severe inflammatory response, and this could affect the clearance of the virus from the serum as compared to naive individuals.
Yeah, that's fine. I'll try and answer quickly. So we're 100% trying to make vaccines now that generate T-cell responses and also an antibody response to the glycoprotein. Our first attempt has been using viral vectors. Not sure viral vectors will be the best way to make an antibody response against glycoprotein, so we're exploring new ways of doing that. But I think everyone thinks the next vaccine we're trying in efficacy testing should be aiming to generate T cells and antibodies. So that's the answer to that question. Your other question about whether vaccines will be effective in people who have had chronic infection is a really important one, because I think that will be a real challenge. Once a patient's been chronically infected, they have an immunotolerant adaptive immune system to the virus. Then you clear the virus with your drugs, you try to vaccinate them. The question is, can you restore an effective adaptive immune response in someone where that's been tolerized because of decades of chronic infection? And I think that will be a difficult challenge. It'll be much better to vaccinate. It'll be much easier and more effective to vaccinate naive people than people that are being chronically infected. Immunologically, that's my predictions, but they're both great questions.
Thank you. Thank you so much.
Thank you. Is there any other question, please? Thank you, Professor Bars. And
thank you very much, and really enjoyed this, and also we are aiming for the collaboration with you and Manal and the hepatitis team in Masri Center. And now is my honor and the pleasure to introduce Dr. Cesar Lopez Camacho. He is holds a PhD in molecular biology from the University of Manchester, United Kingdom, and has held 4 poster positions in molecular biology and also for gene expression and vaccinology at UMass Medical School in USA, University of Vermont, colleague of Medicine USA, and Jenner Institute at University of Oxford. He is currently the mRNA technology program leader at Jenner Institute. His research focus in the following: mRNA-based vaccine development, viral vectored vaccine research, vaccine development of arthropod-borne pathogen, mosquitoes and ticks. For this, he uses genetic design of mRNA vaccine platform, utilizing molecular cloning, in silico gene design and optimization of gene expression with high induction of adaptive immune response in preclinical model. Professor Camacho, will go for breaking barrier innovation in RNA-based vaccine for emerging infectious disease. Thank you very much for coming, and also thank you for the idea for exchange in the morning. So thank you very much, and the floor is yours.
Thank you for inviting me.
This for the next?
One
once the presentation is in. Yeah.
Can you check it here next?
Yeah.
And then previous.
Mhm.
Okay.
Thank you very much for the invitation, Dr. Mahmoud Al-Mitini, for the kind treatment of our staff, Dr. Ahmed, for bringing us from Oxford to Egypt, my first time in Egypt, and I will take it to one of my most impressive and nice travels in all my time. It's been amazing to be here, and I'm overwhelmed by the kindness of you. It's amazing to be here, and it actually reminds me of Mexico. In Mexico, we are also very kind, very friendly, always smiling, and offering tacos to everybody. So one thing I like to start is basically telling you more the story about mRNAs in the genre, how we got to make mRNAs. And the story is that I never believed in mRNA. You know, we've been focusing on the novel vectors for a long time. And the reason I never believed in mRNA technology is because it was too complicated. And now look at me. I'm the group leader of an mRNA-based program. But we'll see, and I'll probably give you an indication that this platform is actually very useful. This is working now? Okay, so I've been in the genre since 2014. I work on vaccine development for arthropod-borne viruses, since dengue, Zika, chikungunya, Mayaro, Vivax, Tycruci, and HPV. So I call it the pre-COVID era. Then the COVID era, when the pandemic struck, I started working with SARS-CoV-2. So the group I was working with, Professor Gavin Scritton's group, and a full army of postdocs, take on in the challenge in producing the spike protein for the first time in the university. We aim to actually produce and purify 100 milligrams of protein to be delivered to many institutions, many universities, many health systems, and that actually helped to standardize a serological assay, between many other things. After the COVID pandemic, I rejoined the genre again. Dr. and Sir Professor Adrian Hill, it's been very supportive in my line of research, and together we have a vision, producing mRNA vaccines and also explore these vaccines in conjunction with our platforms. So you know about Oxford, you know about AstraZeneca, you know about the chimpanzee adenovirus. But perhaps the thing that you might not know is that this vaccine actually saved over 6 million lives. And this is astonishing. It's amazing. And this picture basically reflects all the people who work very hard to get the vaccine in your arms, in our family's arms. And we are very grateful to be part of this effort and all the persons who took part on this. So what will be the future of vaccines then? Oxford AstraZeneca was the starting. These people would think that, but in reality, the genre has worked on vaccine development for many, many years. So in this case, I will focus on an overview of the mRNA anatomy in a very simple way, so you won't get lost. I'm going to explain how is it made, how is it encapsulated. Many of you perhaps have got the vaccines for Pfizer or Moderna. This is a lipid nanoparticle, and inside the lipid nanoparticle, there is the mRNA. So I will talk about the mechanisms of encapsulation and what are the capabilities of the encapsulation in our institute. We'll talk about the thermostability, importance of thermostability, formulation for efficient lipid nanoparticle mRNA delivery. I'm going to take you through some working cases, SARS-CoV-2, malaria, tick and tick-borne pathogen mRNA vaccine development that I'm working with. And finally, some visions and perspectives. So this is perhaps the fastest course of molecular biology you will ever get. So you have a DNA
which is the coding gene, and by a mechanism called transcription, you'll have production of mRNA, and that will basically be the message, and the message will be read by ribosomes, and that is basically known as translation. That translation will generate a protein, and that protein will give you the functionality of the specific molecule. Obviously, this is the most simple way, but there are plenty of books and papers that actually explain the molecular mechanisms of every single step that I just mentioned. But more importantly, we need a ribosome. And the ribosome is a big complex of several proteins that help to read the mRNA and then produce a nascent polypeptide. And this ribosome needs to meet with the mRNA, and you have to give signals to the mRNA and to the ribosomes to meet each other and to start working together. So the anatomy of mRNA is basically very simple yet complex. We have the mRNA is composed first with a chemical modification, which is called the 5' cap, and this modification maximizes the stability and increase efficiency of stability of mRNA, followed by a 5' untranslated region. This enhances translation efficiency, but also promotes stability, followed by a translated region. This is basically what sort of gene you want to express in the mRNA. And this is a big deal because you have to optimize the gene in a way that expresses correctly. Optimization of gene means codon optimization, but also means the depletion of uridine and then the modifications of uridine, which is called the pseudouridine modification. So that's another different and very important complex part of the mRNA anatomy for therapeutics and vaccine development. Then we have a 3' UTR, which is, again, it's an untranslated region. It optimizes and enhances the translation. And I have to say 5' and UTR are actually very important because even from some viruses, if you do a single mutation of the 5' UTRs, viruses are inactivated or they are more attenuated. So it's important the 5' UTR because the 5' UTR will fold in a three-dimensional way. So if there are structures that are detrimental for translation, then the mRNA will not be produced, or the protein will not be produced in that case. Finally, the polyadenylation that enhances stability. So basically what you have is a molecule that will have a shield, which is the capping, and then the poly-A, and that will protect this mRNA signal from enzymes that will eat the mRNA. So you want to actually have all these little components in the vaccine. The other thing is that the dichotomy of mRNA is basically that you are doing work for therapeutics and also work for vaccine development. In one way, you want to induce immune response against the antigen, and for the other one, you actually want to supply a gene, but you don't want to make it immunogenic. So this is an important work and reflection about this technology. Is not straightforward. The capping, as I mentioned, again, is not trivial. It's very difficult to make, very difficult to produce, very difficult to modify. And there's this new technology developed by TriLink, part of Mara Biosciences, that we are actually collaborating with. They are producing new modifications in the 5' cap. It's can you tell me how do I do the pointer? Okay. Okay, anyway. So we have the 5' capping there at the left, and the modifications that TriLink is doing is basically adding methylations on the structure, and this actually increase the transcription yield by threefold. So we're talking about biomanufacturing what you want is to optimize the maximum efficiency of protein, maximum efficiency of mRNA, and therefore you will perhaps have twice or 3 times the doses that you need. So it's important to take in mind that every single part of the anatomy that I mentioned, it has to be carefully designed. Same with polyadenylation. Polyadenylation problem is that you cannot have polyadenylation on a plasmid that is being used for the starting material for mRNA, and the reason for that is that bacteria like to recombine when there is a lot of polyase in their genome. So there are many strategies that avoid the polyadenylation and recombination, and somebody's telling me, I'm good at the moment. That's perfect. Thank you so much. So the poly-A is also important because that is the signal from a polyprotein complex that will actually make a loop that connects the 5' and 3', and that will allow the translation of the mRNA. So how we actually make mRNAs in the Jenner? So we first do plasmid cloning. We synthesize a gene, and this gene is, it will be governed by the T7 promoter. Once you have the plasmid, you do a transcription, and this transcription is called in vitro transcription. That means that the plasmid will be the template, and then you will add the ingredients needed to produce the mRNA. And the next step will be to add in the capping, and these mechanisms can be done within the transcription, within the first reaction of in vitro transcription, or it could be a co-transcriptional capping. These 2 methodologies are important to take into account, especially when you are progressing your vaccine into the manufacturing and GMP. The next step is once you have your capping, then the purification of the mRNA is important. You have to have a pure mRNA. When I mean pure, I mean all of the ingredients that you use for the IVT, they have to be removed, but also making sure that you have one single population of mRNA that is not changing. So you have to have a biologic that will have a consistent amount and length of mRNA that will be functional. You don't want to have a truncated mRNA because that will give you a truncated antigen, and therefore your vaccine or your biologic would be not optimal. So once you have the mRNA produced, the next step is basically to make these small balls of lipids in which the mRNA will be contained inside, and the way this is done is by mixing a specific ratio of lipids. And one most important one is the ionizable lipid, and this is in blue in the section in the bottom. That blue represents the ionizable lipid that will interact with the mRNA, and that interaction will lead later to the conformation and complexing of lipid nanoparticles. And the only way to do that, I mean, you could do it in a very ancient way by dropping and doing some shaking on the tube with the lipids, but we have a more sophisticated technology at the moment, which is called the nanosampler Ignite. This particular machine helps you at using advantages of microfluidic technology and a chip that it's only 1 cartridge per use and per formulation to give you homogeneous lipid nanoparticles. So as you can see in the electron microscopy photo, the size is important. You have to maintain the same size, especially for a biologics point of view, but also you can actually play with the sizes, and I'll tell you more about that one. So, the mechanisms of encapsulation. I always put 2 things. First, the packing. Imagine that you are sending a package to your family, so you first need to pack your message, your gift. And once you pack it, you have to take it to the delivery point. So the mechanisms of encapsulation basically means that you are doing first the packing that then will be helping you to deliver the message. And the delivery, it's important because the LNP will need to be taken by the cells by a specific cellular mechanism called endocytosis using low-density lipoproteins. And once the LNP is actually taken over, then the ionizable lipid sends the pH from the vesicle, pH low, ionizable lipid dissociates from the mRNA gets released, and all the particle gets released to the cytoplasm. So now the mRNA is ready to engage with the ribosome and to start translation. So we applied for some grants a year ago to have this technology set up in the Jenner. So we start from scratch, as you can see before, empty bench, after lipid nanoencapsulator from Precision Biosystems. And that's basically an interesting achievement, because by pressing one button and start encapsulating took us about 1 year. 1 year of planning, 1 year of setting up on our own strategy for in vitro transcription, for setting up all the reagents needed, working out with the confidential disclosure agreements to make sure that we were actually using licensed products that we could use for the preclinical development. And later on, we actually acquired another very nice toy, which is a synthesizer that helps you to actually measure the lipid nanoparticle after formulation. This is a delay. 1, 2, 3. Okay. So we are focusing with working with precision nanosystems in a way that every single mRNA that we do in the Jenner has a SOP, and this SOP can be transferred to GMP or pre-GMP using the next stage of the machines that are useful for the encapsulation of the lipid nanoparticles. We are working as well with novel lipid nanoparticles developed by Neovac, and all these LNPs are compatible with encapsulation protocols in the Jenner, and they are useful for preclinical and GMP manufacturing. So let's go to the science and the results. So this is an example of a mRNA production in the Jenner, and then followed by a LNP encapsulation. So in here, what we did is that we produce a GFP mRNA to use it as a reporter, or either for in vivo purposes or for in vitro purposes. So as you can see on the first top panel, this is a capillary electrophoresis that shows you the purity of your mRNA, followed by an in vitro transfection to show you that the GFP mRNA that we are producing and is encapsulated in our facility is actually working. The other thing that we do is that we test the encapsulation efficiency. So that means how much of the mRNA is encapsulated in the lipid nanoparticle. So as you can see in the right, in the gel for mRNA, we have the two first lanes are controls for molecular markers. The second one is the RNA, which is formulated with and without the detergent. So when you put Triton, the lipid nanoparticle dissolves, and if it dissolves, then you release the mRNA, and that's why you see the shift that you can see there, with or without Triton. We have achieved encapsulations up to 97%. This one, it was 94%, but to be honest, 94% is just perfect for the purposes of what we are doing here. Jesus with this. So how we test the efficiency and the stability of these mRNAs that we are producing. So first of all, it would be test the vaccines and in this case, the mRNA LNP under different conditions. The conditions would be temperature. So we'll keep the formulations at 4 degrees, minus 20 degrees of room temperature, and we follow up 4 different times. That could be starting from week 0 to week 1. In the top panel, week 0, you can see on the normal fluorescent microscopy, you see green cells, but obviously that's not enough if you want to quantify, so you jump into flow cytometry, and as you can see in the panels, in the left panel, it's a control of unrelated mRNA transfected, and then the next one is the transfection with mRNA at 4 degrees, followed by -20 and room temperature. And the same pattern of efficiency of transfection, which is nearly 96% in most of these conditions, allow us to be confident that our formulations are actually quite stable, either in room temperature, 4 degrees, or -20 Celsius. And remember, this was a bit of a problem in Pfizer and Moderna vaccines at the moment. During the pandemic, the problem of the stability and the cold chain of mRNA vaccines. So now things have progressed, technologies have evolved, and now we are able to produce, manufacture in preclinical settings, stable vaccines for several diseases. On the right panel, finally, what we got is the sizing. So how big is your lipid nanoparticle and how it behaves in different conditions. So when you keep it at -20, at 4 degrees, or at room temperature, respectively, you can see the peak that is about 110 nanometers, which is the average size of a lipid nanoparticle encapsulation. And this size is actually, it can be variable depending on the way you actually produce and formulate the vaccine. The next step would be, okay, so how does it change from 20 degrees to 37 degrees? 37 degrees is the temperature we are actually immunizing, or we will be immunizing patients. Well, as you can see, actually, it's even better. 37 degrees in blue in the bottom graph. The sharpness of the homogeneity of these LNPs is astonishing. So how does it work when you actually work with different lipid nanoparticles with different sizes? Using the same mRNA, the same coding mRNA, in this case, a spike protein from COVID. 4 different candidates, all of these 4 different candidates working differently in terms of size of the lipid nanoparticle. So we prime mice. 2 weeks later, we collect blood. 1 day later, we boost again. 2 weeks later, we collect the blood. So we have 2 measurements, 2 reads, 2 plasma, and then we do an ELISA. And ELISA is basically an assay that helps you to determine the binding of antibodies against your specific antigen, in this case, a spike. So as you can see in vaccine 1, 2, 3, and 4, blue is prime, red lines is boost. Every single line is 1 mice. And as you can see, there is a variation of responses coming from the prime, but mostly all of them catching up in the boost. So whether or not we have a difference of LNP size does not affect the quality of the overall response in terms of boosting strategy for this specific vaccine. So it is still interesting to actually focus more on how you can actually improve vaccines in terms of immunogenicity, but also we have seen that these vaccines can have an impact depending on the size in terms of biodistribution, which is also an important part of research in the group. The final point of this experiment basically was to define the best lipid nanoparticle to be used in clinical trials in humans. So now we are supporting this, leading by Adrian Hill and Kate Ewer, and I'm helping to do the preclinical stability testing for this. Hopefully a clinical trial will be on track for primary objectives. Objectives would be safety and immunogenicity. Secondary objectives, the effect on liver enzymes. And the volunteers is going to be 36 healthy volunteers. Next example, malaria. Adrian will be speaking to you more about that, but Dr. Hamed has done a good introduction in terms of malaria and vaccines for malaria, in this case R21. Interestingly, Malaria Day was on the 25th of April, very recently. I don't have to go through all this introduction, as you have heard this in the previous talks, and I'm pretty sure Adrian will be also mentioning about the good news about this vaccine. But then the question is, can we use this technology with mRNA for malaria vaccines? And this work is basically supported by collaborators, but more importantly, supported by Adrian Hill and Dr. Ahmed Salman, who is inputting a lot of knowledge in malaria biology, helping a lot in the design, and we are doing together this experiment. So what I'm telling you now is that what we're making is mRNA vaccines expressing the antigen for malaria, which is called the CSP, you remember. And remember as well that R21 is a heavy surface antigen VLP-based vaccine. So we are actually focusing on two strategies, making an mRNA vaccine that is expressing only the CSP, the orange part on the outside of the particle, but also making an mRNA vaccine that hopefully will express heavy surface antigen followed by CSP and hopefully making VLPs in vivo. So as I mentioned, the design of a mRNA is complex. You have to deplete uridine, you have to do a lot of bioinformatical analysis to make sure that the structure and the three-dimensional structure of the mRNA is not giving you loops that are detrimental to mRNA. So that's important to consider. Here in the bottom part, we show the purity of our mRNA production and then the encapsulation efficiency and the right size of lipid nanoparticles. Same with the 96% encapsulation efficiency. So are our vaccines that we are producing from scratch in the genre immunogenic? Yes or no? So we are here presenting only one part of the story. It is very recent data. We produce an mRNA-based vaccine expressing only CSP. Remember, this CSP is only the external part of R21. And remember that this vaccine is unadjuvanted. R21 needs matrix M to be effective. So bear in mind that. So in here, interesting experiment, immunogenicity, inject mice, 3 weeks later, boost the mice, collect the sera before the boost, obviously, and after 3 weeks, bleed the mice again, and then do a challenge, intravenous challenge of malaria transgenic parasites. The graphs that you see below is showing the prime and the boost of the vaccines in blue mRNA CSP and in red R21 matrix M. So in terms of priming, the responses are positive. So we are very happy we have immunogenicity specific to NAMP ELISA. And also after the boost, we are inducing immune responses specific to CSP. So in the top right of the panel, you can see the comparison of the endpoint titers of these vaccines, comparison between mRNA and R21. For prime, the sera dilution is 2.8, 3.7. 3.7 is red, so R21 matrix M is still surpassing the effectivity, and it's more immunogenic than mRNA CSP. Followed by the boost as well. Both of the vaccines boosted the animals. However, mRNA vaccines were able to boost to the levels of the prime that you were achieving in R21 vaccine. So the question is, can we increase the levels of immunogenicity for mRNA? The answer might be yes. We will be doing a second experiment, doing a third immunization with a CSP mRNA. But remember as well that we are not using an adjuvant. So it's interesting to note that. We need to take in consideration that a second boost can increase the antibody titers. And you also have to remember that we're still working on getting data for the mRNA expressing the whole Hep B surface antigen fused with the CSP. In the table, finally, this is just the early stages of the challenge. At day 5, we have the groups, group 2, 3, and 4. Group 2 is mRNA CSP. group 3 R21 matrix M, and then naive mice. So we see a slight changes in the parasitemia. So parasitemia, that means 1% parasitemia, you consider that the vaccine didn't work. So when you progress to 1%, you have to cool the animals. So it seems that R21 matrix M is still doing their job as it should be. And we have some a slight evidence that there is some delay in the challenge, but we have to follow up and do more experiments and to do more statistical analysis. The last example of what we are doing in the journal is developing mRNA vaccines against tick-borne pathogens, but more importantly, against the tick itself. And that means that what you want is to induce immune response against internal regions of the ticks, and that will hopefully produce anti-immune response, and you will perhaps feel that the tick is biting you and you probably remove, or there's another strategy. So in this case, I am working in conjunction with I guess one of the most famous tick-borne pathologists and tick biologists in the world, which is de la Fuente, Jose de la Fuente. He's in Spain, but he's actually Cuban. And he has worked many, many years on two candidates, subolecin and MSP4. Subolecin is an internal mid-gut protein that is important for reproduction, development, and behavior. And MSP4 is also a mid-gut that is a protein located in the ticks. So when you inject cattle or rabbits or rats or whatever mammal, you're going to get responses anti-subolecin, anti-MSP4. When the ticks feed on these vaccinated animals, the blood contains the antibodies against subolecin and MSP4, and they impair the feeding of the tick, and the tick fall off detached from the host. So what you want actually is to control and to avoid the tick infestation, but on the other hand, you also want to vaccinate against a specific tick-borne pathogen. So this is just a starting point for the dual mRNA anti-tick vaccines. We are taking these two vaccines. Previously, they've been using as a subunit vaccines, but now we're actually using them in a dual anti-mRNA vaccines. We are now progressing to preclinical models as we speak, and we are sending some of the sera for function analysis in the next few months. I will skip this. And the summary of this talk is basically that mRNA, I see it as an engine of a car. So if you want to decide to engage into mRNA, you might decide to work on an mRNA from an engine, from a Volkswagen, or from a high sport car, Ferrari maybe. And the complexity will increase, but it will pay off. And this is the way I see when I work with mRNAs. Every single person that works on this has a role. And it's not only me, it's all the group. And basically, we are hiring. We are accepting the field masters, students, fellowship, postdocs, etc., etc. And I wanted to bring up this opportunity for students like you guys. The Nuffield Department of Medicine offer at the moment a summer studentship program. This gives you the opportunity to come to Oxford and experience research for 2 months during this summer. The call is open. Please apply. There are 10 different projects that you could choose. I'm actually offering one in terms of mRNA vaccine development. Just so you can have a picture on this, have a look, and probably apply if you wish to. Finally, thank you to all the persons involved in this great seminar. Thank you to Ahmed to helping me to bring all these new ideas and results to you. Thank you for the funders. And this is the actual people who is making the work. William Taylor is a French postdoc that joined 4 months ago to our group. Adam Truby is our research assistant, a wonderful person working and helping us with the preclinical assessment of the vaccines. And our 2 students, Celeste and Rebecca, that are starting to experience science. So I will be happy to take any questions. I'll leave you with this very nice verse regarding vaccines. I really enjoy this, and I hope that you can actually come to Oxford at some point. And I'm happy to take any questions. If not now, later.
Really, thank you very much for this very, very impressive lecture, and I think we learned a lot from all of these, and I think there's a lot of questions, maybe, I think, yes. I will start with His Excellency Professor Dr. Awad Tajuddin, if he want to comment about the vaccines and the development of messenger RNA versus live attenuated. So, Professor Awad Tajuddin, he is the advisor for the President of Arab Republic of Egypt for the prevention and treatment, and he is a professor of pulmonology. So he is involved for the elimination of the COVID in the COVID era. So, Professor Awad, please.
Thank you very much, Professor Hisham. Just I'm coming now because I have a meeting with the President all over the day. But I hear the last comment, which is very important, so that we appreciate your comment about the Egyptian peoples and the old Egyptian people, which is true. But from my specialty, I'm pulmonologist, as I mentioned yesterday, so that the vaccines for us are very important. And still we are waiting for the new BCG vaccine or new anti-tuberculosis vaccine to get a strong, long-term actual protection against mycobacterial infection. Fortunately enough, we have a very good achievement in the management of tuberculosis. We achieved now the prevalence of tuberculosis in Egypt is 10 new cases for 100,000 population. And about 30 years ago, the number was about 300. So that if you compare Egypt with different countries in the region or in Africa, this is a very good achievement. We have a very strong program against tuberculosis. The diagnosis and treatment is free of charge for all peoples and even for immigrants here in Egypt. And the drugs, including the medication, some of the drugs are produced here. And we have facilities for tuberculosis long-standing. This is about Egypt. We have 38 chest hospitals, which was called sanitarium before, and now we have dispensaries distributed all over the country for diagnosis and treatment of tuberculosis. The already present BCG vaccine is obligatory for all children since birth, but as you know, BCG vaccine, which is available now, it gives protection against the fulminating forms of tuberculosis as miliary tuberculosis or tuberculous meningitis. But we hope for the near future we have a new vaccine for tuberculosis. The obligatory vaccination in Egypt is for many diseases, and now we introduced in Egypt for this year, 2023, the anti-pneumococcal vaccine, and Haemophilus influenza vaccine now is obligatory, so that the program of vaccination in Egypt is a very strong program. I think there is pressure from companies producing the vaccine to reduce the cancer cervix vaccine, and we hope to introduce this vaccine next year. Thank you very much.
Thank you very much. Yes.
So, another question? Yes? So, we have a lot of questions.
Thank you very much for your nice presentation. I used to say that the only winner of the COVID-19 era is RNA, because what was taboo before is now under license, and the application of RNA in therapy and in vaccination after the COVID is now very open wide. My main concern is the biosafety or the We learned from the RNA interference that introduction of exogenous genetic material inside the living cell can lead to a cascade of events or cascade of reactions that we don't know the outcome of it. What's your opinion about the biosafety of using RNA in case of vaccination or RNA therapy, and how we can deal with that, how we can prevent that?
I agree totally with your comment. I guess it's important to hurry, but to walk the path in the right way. I think it is important to boost the funding in terms of basic research. So we know that mRNA is a good technology for vaccine and for therapeutics, but we are in a way forgetting that mRNA biology has been there since 70s. So when I go to the 70s papers and I read the papers, this is just like gold to me, because you see all that's been done that you can actually apply to the vaccine development, and if you do that, and you have the time to actually work in a better way to improve vaccines, is best. So coming back to the question, I guess it is important to know that mRNA on its own has a half-life. It won't stay for long, and it won't have a big effect as siRNA, for example, in which you can have off-target effects. Also, that would be a problem. But I agree with you, in terms of therapeutics, there is a lot of things to be done. And the only way to overcome this is by doing research and focusing on these important questions for, DPhil projects on the master students and so on.
Thank you.
Okay, thank you very much. I know there is a lot of questions, but Dr. Salman said that we are running out of time. So, please, okay. So, His Excellency the President, last questions. I'm sorry. Okay.
Thank you so much for this nice presentation. As an expert in this field, I have two technical questions. First, my name is Amr Abdel Hamid. I graduated from Faculty of Science at Shams University Biochemistry Department, and now I'm a pre-master student in Biochemistry Department, and now I'm teaching assistant at Faculty of Education, Chemistry Department. The first question is about stability of this molecule. As you mentioned, Moderna and Pfizer vaccines are based on the same idea, the same concept. However, the two vaccines have different, storage conditions, right? For instance, Pfizer vaccines should be kept at minus 80 degrees for 5 days maximum, and Moderna vaccine, it can stay for 30 days, and it can be kept at minus 20 degrees Celsius. So, in your point of view, what are the factors that affect stability of this molecule, messenger RNA?
Okay, so thank you for the question. To be brief, you need a right formulation. So right formulation means the buffers that you're using for the vaccine. It has to be taken care, because if your formulation is not suitable for freezing conditions, that might be detrimental. Second is that every single molecule of mRNA is different. I think I showed you some of the three-dimensional structures of this. So every single molecule that you make has to be carefully taken care in terms of the stability. It's not one formula for all. So there is going to be a lot of research and development into every single vaccine that you produce. But if you have other questions, I'm pretty sure we will talk, because we are actually in a rush. Yeah.
Thank you so much again, Dr. Cesar. It's my pleasure to introduce Dr. Adel Turabi. He has nearly 20 years experience in developing VLB vaccines and nanoparticles. And he actually merged between both industry and academia that make him really expert to understand how to link the bench experience to industry and to make a product. And he's now leading, I will call it, the non-communicable disease vaccines, or I call it sometimes the crazy vaccines. So he will talk to us today about his expertise with that. Thank you, Dr. Adel, for being with us.
I didn't hear my own introduction, sorry. Well, assalamu alaikum wa rahmatullahi wa barakatuh. Thank you very much for the invitation. It's a great pleasure to be here. And I think, like everyone, I'd like to thank all the organizers and especially you, the audience, for coming and listening to these talks. I'm going to dive straight in. So our group is working with vaccines, but we're targeting the non-communicable diseases. There's only a few of those, so we've got an easy job, right? The idea here is to not reinvent the wheel. As you can see, there's lots of different areas, and infectious diseases is important, but actually the majority of morbidity in the world is through chronic diseases. So you'll see on there a lot of targets which you wouldn't necessarily think are vaccine targets, correct? Okay. And, you know, we talk about health. Oh, thank you so much, sorry. We talk about health and disease as like it's a thing, but actually it's a journey. We're always some degree of health, some degree of disease. And I guess it's really poor show for me as a non-medic to try to lecture to medics, you know, how to deal with this. But, you know, the reality is the struggles are we are living with limited resources. There's not a healthcare budget big enough to deal with it. And with our successes, we create a problem for ourselves because aging is a major risk for chronic diseases. And, you know, this is one of the global challenges we have. Our population globally is growing, and the largest increases are seen in the developing world. So by the 2050, we were going to have an extra 2 billion people over the age of 65, which is fantastic, but also brings with it challenges if we want to treat these diseases that come with old age. And these diseases usually are not cheap. There has been a revolution in the use of these new age molecules, these biologic drugs. I'm probably the most well-known of these are the monoclonal antibody therapies, which you will know. And they're treating a lot of the diseases. And you can see over the past 30 years, 40 years, there's been a massive increase in the numbers of biologics in development. And just you can see on the right-hand side, you know, a large proportion of those are actually targeting the cancers, but also the non-cancers. What do I want to say about this? This is great news because medicine for illness means there's hope. The other thing it says is that here's an immune homologue, this antibody-based therapy, which can target diseases. So if an antibody can make a difference on these diseases, maybe we can recreate a vaccine to do the same. So my premise to you is to look at therapeutic vaccines as an alternative to these passive immunization therapies. And the technology I want to talk about, the technology that I love, is the virus-like particles. And I think we've heard a few of those today, and we're going to hear a bit more in the next speech, because there's a very famous and good malaria vaccine doing well that's based on virus-like particles. So just very quickly, these are recombinant proteins that spontaneously self-assemble. So this is the best part of it. You just make it, and then it will do the rest. And the other thing is, they're non-infectious. This is an important, so especially if you're going to be treating vulnerable patient populations. And whenever we can learn from nature, we do well to do that. Viruses have been around for a very long time, and they've learned to be very economical with their genomes. And so if we look at this simple building brick, you can see that you can manufacture very great and amazing structures. I was going to put a pyramid on here, but I thought that would be too cliched. So by exploiting this architecture of viruses, we can express just a single protein. It will self-assemble, encapsulating RNA or DNA, and creating a virus capsid that is non-infectious. There are more complex assemblies that use multiple proteins to make up the capsimeres, so such as the picornaviruses. But all of these are just basically showing you that we don't need to reinvent the wheel, but we can reinvent the wheel as well. VLPs are familiar. We know the Hep B vaccine is a virus-like particle, so you've probably all had that. We also have this HPV. It's a very successful vaccine. Originally it was 3 doses. Now it's a single dose can give as much protection. So it's really interesting. They're highly immunogenic molecules. I always like putting up this slide because I figure by this time of day you're feeling hungry. And so the reality is we have as many flavors of virus-like particles as there are viruses. And you know, the thing to do is to select whether you need, if the antigen is particularly complex or what kind of expression system is best used for your VLP. And what I want to highlight, I'm not sure if it, yes it does, okay. So the superpower of these systems is that the particles themselves, you can display a second antigen on there, and so a heterologous antigen onto those particles, and that's a superpower, I think. So where do we start? We start by choosing a good scaffold, and the scaffold is fit for purpose. So on the left-hand side, we have this bacteriophage, Q beta. This is by far away, like when I started working with this, I cried with joy. It's so easy to make. It can be made in gram per liter quantities. Now this is important. And it's almost bulletproof. We had a batch made in 2007, and it just sat in the fridge, and and I came along in 2014 and was using it, and we used it, and I even gave some to Ahmed. We used, and it's like bulletproof. So these things are incredibly stable, and this tells you something that, you know, when a virus gets it right, it gets it really right. So we can hopefully, you know, get away without this ultra cold cold chain storage with these particles. On the right-hand side, we have a bunch of other virus-like particles we work with, some bacteriophage. The one you'll see there says cucumber mosaic virus. That's the one I'm working with as well. Actually, it was brought out of a tiger lily, but we'll continue. And then in the middle, at the bottom here, you can see the very famous Hep B surface antigen. There's lots of VLPs. What they all have in common is that they're beautiful. So what makes them good as vaccines? Number one, I've said it, safety. They're safe to manufacture. We don't need the virus to make this. We just take the gene for the protein and put it into an expression vector. The vaccines themselves, without having that replicative machinery, without having that infectious material, means that from a regulatory standpoint, they are a no-brainer, and therefore we can administer to a broader range of patients. Number two, they are the optimal size. Viruses we've lived with all our lives, as Professor Barnes introduced this morning, and so our immune systems are highly prejudiced against them. So these virus-like particles are all of this nano scale, this 20 to 100 nanometers normally. Some larger ones are 200 nanometers. That means that they effectively drain to the lymph. The lymphatic tissue is obviously where all the good immune cells are just waiting to be educated. It's like the university of immune cells. And then also they're not too big so that they are effectively taken up by cells and processed. So they sit in this golden size range. The other thing is they look like a virus. They look dangerous. The immune system finds it hard to ignore something like this. So this repetitive surface is a feature of pathogens and will induce the immune system. They're called PAMPs or PASPs. This repetitiveness also can induce cross-linked B-cell receptors and therefore kick off in B-cell stimulation in a T-cell independent fashion. This is just to get you going. It's like a starter, an appetizer. They also can bind all the other components, active complement system, natural antibodies. And then the last thing, which I think is really important, is that they encapsulate. You can put stuff in them. And in this case, we're looking at just their native RNA or DNA. But those molecules themselves are potent immunostimulators, and they will switch on pattern recognition receptors, such as the toll-like receptors, which are then... what this means is that these virus-like particles, these ones that carry the RNAs, are very immunogenic without actually having to add an adjuvant. Hep B surface antigen needs an adjuvant because it doesn't package any DNA or RNA, other reasons as well. Okay, let's see if this... So, how to adapt the VLP for making your vaccine. So, if we start with your design, you'll look at your target, maybe do an omics or look at structural aspects of it, use bioinformatics to kind of narrow things down, use the systems immunology approach. There's lots of ways to go about it, but let's just skip this. You then choose your scaffold, and then you can also then go ahead and engineer it. So in the case of chronic non-communicable diseases, many times we're targeting our own antigens, these self-antigens. I've just listed here a few that we've worked with with a group in Oxford, ranging from Parkinson's, psoriasis, asthma. I'm not sure I'm going to have enough time to actually go through these because I want to wrap up and get to the next speaker. But the real thing that I want to show is that each one of these targets, there's the protein alpha-synuclein, the cytokine IL-17, nerve growth factor. There are monoclonal antibodies in clinical trials that are validating them as biological targets. And those clinical trials kind of give us the confidence to think, okay, this is something we can intercept with. On the right-hand side, you'll just see this very simple cartoon of how we do things, but I'll come to that in a second. The other thing is, I talked about an aging population, and what I wanted to say is that we can also adapt these virus-like particles. We've taken our cucumber mosaic virus, and we've fused onto there the tetanus toxin T-cell epitope. And what that means is, as you all have received, I think we're roughly around about 96 to 99% immunized with tetanus. So everyone has a memory T-cell response against tetanus. And therefore, by incorporating this epitope in our vaccines, this is another way of helping people with less active immune systems to create immune responses. This itself is a whole talk by itself, but I'm going to pass. Then, once you have this beautiful optimized particle, you then decorate it. I call it the dark art because actually this is where it gets really, this is the difference. How well you present things on a particle is directly correlated to how good the response is. So the quality of the resultant antibodies really are dependent on how authentic the antigen is you put on it, the orientation and all those other things, the epitope density, how much you actually stick on, and then also the stability of your antigens on the particle. And this is all very important to think about because this all has a bearing on manufacturability, which has a bearing on deliverability. Ultimately, someone has to pay for this, and that someone is us. So the first decorative method I'd like to introduce you to is the simplest, where you take, you just do a genetic fusion of your antigen onto to the subunit of the virus-like particle. You can then go more exotic and make what I call these, so instead of having just a chimeric molecule, you create a mosaic. So for some proteins, when you introduce an antigen, won't assemble properly. So the way to get around that is to incorporate some of the undoctored subunits. This was the case with the first malaria vaccine, where they tried to make a chimera and they weren't able to. So they basically then had to make 1 part chimera to 3 parts native Hep B surface antigen, and that was the RTSS that we heard about yesterday. The other decorative method is what I call stick it on. Bioconjugation. So we just we look at surface exposed reactive groups on the surface. You take a chemical cross-linker, and then you stick your antigen via that chemical cross-linker. And this creates a very nice, highly repetitive decoration. As I said, it's very versatile. There's, you don't need to mess around with your antigen too much. You just need to maybe think about where you're going to put like a free cysteine or another acceptor molecule on your antigen to get it to cross-link effectively. Lots of different chemistries are available, and I love this stuff. I could talk about it forever. But really, all it is, you have a plate, and you're just deciding what cupcake to put on there. Proof of the pudding. Okay, so I'm going to now talk about a few examples. So I'm not going to introduce psoriasis. You all know about psoriasis. But just to say that, so the targets there are these cytokine. And so in particular, this IL-17 was shown to be very effective. So we tried to make a therapeutic vaccine targeting IL-17. And we tested this in this model, this mouse model, where we have our vaccine with the IL-17 on it. We immunize the mice, and then we stimulate psoriasis-like condition by using imiquimod. It's a TLR7 agonist, and that creates the epidermis thickening that you see. And if you measure that imiquimod, so the red bar, you can see that the epidermis goes, so untreated versus the treated, you can see that the imiquimod is thickening the skin. And then if you add an antibody that blocks IL-17, it reduces it. Now, what was really pleasing is that with our vaccine, we were able to match the efficacy of the antibody treatment just using a vaccine. And, so this kind of suggests that you can actually target these pro-inflammatory cytokines very effectively with a vaccine. And this is just to show you, this is an arm of a person with psoriasis before treatment. This is not our vaccine, by the way, sorry, I should say. This is the antibody treatment. But it's just to show you how effective blocking IL-17 is and so after 4 weeks, they've gone from this very severe to very mild. And within 12 weeks, they're fully clear. So, but, at a cost of several £1,000s per patient per annum, it's going to be a costly job to do it. So with our vaccine, we can match the efficacy, but is it safe? So what we did is we tested this by inducing endogenous IL-17 using imiquimod, so where the arrow is indicating, and what we're looking for is to see if there's any increase in IL-17 in the if we can induce anti-IL-17 antibodies. Have we broken tolerance by giving this vaccine? And the answer is no. You see that the antibodies for IL-17 stay flat after doing this imiquimod treatment. But when we use the vaccine, you can see that we get a re-stimulation of the antibodies. So only in the presence of the vaccine, only when the IL-17 is on the vaccine, do we actually create an anti-IL-17. And that means that we're not breaking tolerance. We're bypassing tolerance. And I just want to, spend a little bit of time just to remind you that tolerance is important in life. We do not want to stimulate autoimmune responses. We want to trigger an antibody that is going to be biologically relevant to the disease, but then when it's done its business, to go away and for us not to have to worry about it again. The biologics have a half-life of between 28, 35 days. They can do stuff to extend them. With our vaccines, our half-life is probably around about the 90-day mark. So it means good thing is that it means there's less dosing, but if it's bad, then you've got to live with it longer. But what's happening with tolerance is, we don't have any self-reactive T cells. They're deleted during development. So without that T cell repertoire, there's no activation there's no stimulation of the B cells in the presence of those antigens. So to get around that, we introduce a foreign T cell epitope, which is the VLP vaccine. And by that way, we are able to stimulate in the same niche active T cells and the B cells that recognize the antigen in the same niche, and then you get antibodies produced. So only in that. Okay, that's it. I've said that twice. So another example of what Ahmed calls crazy vaccine. Pain. Pain is not a disease, but it is absolutely one of the most prevalent conditions, and costs perhaps more. I mean, this I was shocked by. It costs more than heart disease, cancer, and diabetes put together in the developing world, in the United States. So there's a very good target for pain, which is nerve growth factor. There's this clinical trial by Pfizer and Eli Lilly. They were using it in osteoarthritis. I'm going to gloss over it, but basically that trial didn't fail because it didn't get rid of the pain. It was some other reason. But all that does is it blocks the nerve growth factor binding to its receptor and reducing the sensitization of these peripheral nerves. So in a non-load-bearing situation, this actually might be very favorable. And we're trying now to adapt this vaccine to treat non-load-bearing pain, like chronic pain, pelvic pain, which is afflicting women with endometriosis. We think there's not enough research really geared towards women's health, and we kind of think that this is something we can address. Anyway, made our vaccine, stick the IL-17 on. This is just some graphics as to how it works. You can see on the right-hand side that if we try and inject the nerve growth factor by itself, there's no antibodies against nerve growth factor. Only in the vaccine do we get antibodies. So then we need a model to test the pain. And so up steps our favorite model, the mouse. and we gave the mouse osteoarthritis by destabilizing the medial meniscus, so a little meniscectomy, I guess, a football injury for mice. And what you can see over the course of 12 weeks that they develop the symptoms of osteoarthritis, and this is through this sum scores, but also if we measure their pain, and this is a really neat contraption because mice don't answer the questionnaires when we ask them. So we put them on this contraption and it measures their weight bearing on each of their legs. So we operate on one leg and the other leg is unoperated. And if you test that asymmetry, you can see that as the disease starts to go up, the asymmetry increases. So the mice start to feel a bit of pain. It's actually not a very severe pain. And if I was in the room, because of my testosterone, those mice, they get a bit anxious and they just feel nothing. So I can't be in the room when these measurements are taken. I have too much testosterone. So with me out of the room, I do the vaccinations and that's it. But working with very good colleagues in the Kennedy Institute, we did this study. So I'm just going to cut straight to, so you vaccinate, you get antibodies, but let me just cut straight to the chase. Here we have two types of phase. There's this post-operative pain, which is an acute pain. So what you can see, we have a mock vaccinated, which is just the VLP, and we have the vaccine candidate. And here, with the vaccine candidate mouse, after 48 hours, they've now reverted back to their non-pain behavior, whereas the mock vaccinated mice took 1 week to get back to their. So in an acute setting, this blocking of nerve, this antibodies induced by our vaccine, were able to block that acute pain behavior in mice. And then we have this later stage. So what you can see, as the disease goes on, the thing gets more severe. So at this point, we do a boost at day 87, and you can see that our antibody titers are going up, but also our pain symptoms are alleviating. So our antibodies, again, so this vaccine is able to induce antibodies that are alleviating chronic pain in this model. Really, this is the best result I've seen in years of doing stuff. And we're taking this forward, and we would like to put it into humans, but we just can't get the funding. So we're now doing this in the companion animals, because do you know dogs have a monoclonal antibody therapy? It's $1,000 per shot, and people pay for that. So yeah, our vaccine will be a lot cheaper. I'm going to very quickly go through this other project. So along with all the other stuff we do, we're also trying to solve dementia. Why not? And so we have this project from the Joint Program for Neurodegenerative Disease. It's a EU consortium that I lead with Professor Martin Bachmann, and we're targeting, it's called OligoFit, and what it means is we're targeting these aggregate species, these mobile aggregate species called oligomers, and we're trying to screen them and try to understand whether we can create individualized therapies to target those neurodegenerative diseases. This is our team. We're basically in universities across Europe, from the east, from Latvia, Poland, Denmark, all the way, Germany, all the way across to Portugal. And whenever we can, we try and meet in Portugal. It's a very beautiful country. So I'm going to skip this one, but basically what we're trying to do is understand how one protein is giving rise to many different diseases. But so these oligomers, they're made from a single protein called alpha-synuclein, and this is implicated in these synucleinopathies, of which Parkinson's disease is the most well-known, but there's also multiple system atrophy, and there's also Parkinson's with dementia and Lewy body disease. And although we say oligomers, there's actually multiple forms, and the question mark is, do these different forms convey different diseases? And there's now evidence, as of a few years ago, that there are maybe different strains for the different diseases. And on the right-hand side, there's just this blot where you can see that there are two different antibodies, one that sees all of them the same, whereas the FILA5 is seeing the PD, the antibody is seeing the PD more than the MSA. So we're trying to take this forward and create new vaccines that will target specific serotypes, strains of these aggregates. So that's the goal. And then what we want to do with that is not actually make a vaccine in the first instance. We actually want to sort of solve a problem on detection. The way we detect disease, so neurodegenerative disease takes decades to manifest. And what we need to do with those diseases is treat them early. And so the way they're doing it is trying to find evidence of these aggregates in the peripheral tissues, such as the cerebrospinal fluid. And so they've developed these seed amplification assays to try and detect them. It takes something like 5 days for a signal to come up. There are problems because of reproducibility and the quality of the reagents. The field is really in need of some standardization. But 5 days for a neurodegenerative disease is not a bad time to wait for a diagnosis, but just saying. More interestingly, that we've seen recently, there's this cohort of individuals who have sleep behavior disorder, and they convert to Parkinson's with very high percentages within 15 years. So we can now we've identified a population where we can test interventions. But right now, what we're trying to do is use our vaccines to make antibodies that we can then put into biosensors and create a rapid point-of-care testing that will be able to differentiate the different diseases. So this is just some early work where we've done this with the biosensors. Now, the importance of this is that is the sensitivity. These aggregates are present in femtomolar quantities, really low quantities. So you need a very high sensitivity. ELISAs can't get down to those kind of concentrations, but with this immunosensor and biosensor type of technology, you can. And so just to say, although we're vaccine people, we're making vaccines to create tools that will be able to kind of develop tools. And then once we've got that information, we can go back and say, well, that vaccine recognized it, maybe we can treat it therapeutically. But yeah, so it's sort of, and I just wanted to show why those vaccines work. So this is me just displaying what avidity and affinity is all about. So a strong binding, basically, monoclonal antibody has one shot to hit very tightly. And so that's what's stopping me falling off this cliff. However, you can also have strong binding if you have multiple sites. So what our vaccine for Parkinson's does is it creates what I call rubbish antibodies, because they can't bind to anything when it's soluble, because the soluble protein is perhaps doing a job. But the aggregates, those bad guys, we want to get rid of. And so the avidity effect comes in. And so basically this low affinity antibody actually is useful. So, peanuts. We also do make peanut allergy vaccines. I wish I could tell you a lot more about this, but I will say that this is actually a success story. It's using the cucumber mosaic virus platform, and this is now going into phase 1 clinical trials in people. So, but it's this whole paradigm shift where we're trying to create an IgG response to skew IgE to IgG to kind of bring things across. So there's a competition element, there's an inhibition through other receptors, and you know what? If you were really interested, find me in the break. But it's a fantastic paradigm shifting thing. We do so many different vaccines and projects. I struggle to try and keep it less than 30 minutes, and I failed. Sorry, Ahmed, I'm really sorry. But this is just some of the selection of our studies that we've done, which I've mentioned. And we've got lots more published examples. I should have introduced us properly. Our group is Martin Bachmann. He's the PI. I'm just the proxy PI. He's based in Switzerland, but we also have labs in China. We were in Qatar for a brief period of time as well. But now I'm fully in Oxford, by the way. Thank you very much. Don't ask too many questions. We're late.
Thank you so much, Dr. Adel, for the very interesting talk and telling us a lot about your crazy vaccines, which is really non-traditional. And that's personally changed my principles and thoughts about vaccines when I started working on vaccine field. I'll be happy to take any questions.
I just want to thank you very much, because I may be one of your customers, because I have a cluster headache and also I have a peanut allergy. So thank you very much. So I feel that is very, very interesting, and also it is important to ask our neurology group to combine this activity. I see all parts of the world except Egypt, so I'm trying to convince you to have
Please. No, I have to say this. One of the things I'm really concerned about is there's not enough diversity. We look at these diseases, and they're first world diseases. There's no such thing. I'm sorry, people have these chronic diseases everywhere. Dementia is a problem everywhere. You know, it's, so thank you, yeah.
Great, thank you. Any more questions?
When's lunch? No, we have a great talk next.
Okay, thank you so much.
Thank you very much.
Dr. Adel.
So now it's really an honor to introduce Professor Sir Adrian Hell. I want you all to welcome Professor Sir Adrian Hell here, please. Professor Sir Adrian Hell is the Lexman Mitchell Professor of Vaccinology and Director of the Jenner Institute at Oxford University. Currently, malaria vaccine has shown the high efficacy in clinical trial in the UK and Africa and is likely to be the first widely used vaccine to impact the great disease burden of malaria in Africa. In 2005, he founded the Jenner Institute at Oxford, an initiative aimed at accelerating public sector vaccine development for a range of infectious diseases, and partnered with the Bridget Institute on Veterinary Vaccine Development. The Jenner Institute is now the largest academic vaccine center globally with clinical stage vaccine programs against 13 diseases. Recently, the Jenner Institute has initiated a major effort toward the rapid development of COVID-19 vaccine, a clinical trial of which he is the Oxford preclinical investigator in progress. He has published more than 600 research papers and over than 68,000 citations and co-founded several vaccine companies. Sir Adrian Hell, please, thank you very much for coming, and we are really honored to have you here.
Well, thank you very much. It's been a pleasure to be here, albeit briefly, and particularly with so many of my colleagues. And what I'd like to do in the next 45 minutes is briefly touch on three subjects. One is how we structure the Jenner Institute, in case you'd like to start your own vaccine institute here. And then tell you two brief stories, both of which you'll have heard a bit about, developing a vaccine for COVID-19, and most recently, producing a licensed vaccine for malaria. So, this is our founder. He lived a long time ago. In fact, he died 200 years ago this year, Edward Jenner, vaccinating his gardener's son to protect him against smallpox. Now, it's fair to say Edward didn't know exactly what he was doing. Nobody knew what a virus was in those days, but he did do the first vaccination that really worked. And more than that, what we're really impressed by was this vaccine went global in 1798. In 1800, when France, as usual, was fighting England, Napoleon stopped the war, so both sides could vaccinate their men. They were men in those days. And vaccines suddenly caught on in a big way. At the Jenner Institute, which is called after him, we do kind of three things. Global health, you'll have seen, we're interested in diseases like malaria and COVID-19 that are of global importance. Obviously, translational research, we're not doing this just to produce papers, we want to produce impact. And we also work with veterinary colleagues in some of our programs, a small number, in what's called a One Health approach, where veterinarians and medics get together to try and work against diseases that infect both humans and other animals. And I'll touch on that briefly with one or two examples. This is our pre-COVID vaccine pipeline in, if you like, late 2019. Malaria has been our biggest program over the years. As you can see in blue here, 24 different vaccine candidates made to GMP that went into arms. A very impressive tuberculosis vaccine program, still going very strong. Hepatitis C, you've already heard about from Ellie Barnes. HIV, a great challenge, very difficult vaccine to make. Pandemic flu, meningitis, RSV, now licensed vaccines in the last few weeks for RSV, not from us, but from major pharma companies, Ebola, and a little bit on cancer, prostate cancer, lung cancer in particular. But as you can see, until then, we had only one vaccine in phase 3 trials. So the major disease themes, and we're almost intrinsically unstructured. If you're an academic at the university and you want to start your own vaccine research program, we're happy to help. We provide core facilities, we provide support, we provide access to our GMP facility if you want to make your vaccine, and so on. So the 3 biggest programs have been HIV, malaria, and TB. That's where we started back in 2005. Recently, lots of interest in outbreak pathogens, particularly from 2014, when we were asked to help with the Ebola outbreak in West Africa and tested a vaccine for Ebola very quickly. Bacterial pathogens, childhood viruses, exciting programs on, say, EBV at the moment, and a single-dose rabies vaccine. And we're having a look at therapeutic vaccines, which frankly are very difficult. If you want to make a cancer vaccine that works, you'll be the first person to do so. That would be fantastically important, but not particularly easy. But I think that probably is the future of vaccines. And I tell the students, if I were giving this talk in 30 years' time, and please God I won't be, the talk would mainly be about therapeutic vaccines, not about prophylactic vaccines. That's the way the world and the field is moving. Just a glimpse at the outbreak pathogens that we are interested in. Essentially, a large program in Oxford, headed by Sarah Gilbert, has made vaccines against all of these pathogens. They've all gone into clinical trials now, including Nipah and Lassa. Some of them have been out-licensed. I won't talk about those in detail, except to mention that the one you might be interested in is Rift Valley fever, which is one of those One Health viruses which affects livestock and people and comes in outbreaks and comes to Egypt occasionally, I understand. 5 large outbreaks in humans as well as livestock over the last 40 years, and no good vaccine, certainly not for humans at the moment. And I think many of these are easier vaccines to make. They're not like HIV. Making a Zika vaccine is pretty straightforward, and lots of people have done that. It's just today there's no Zika around to test any of those vaccines, which is good for the world and bad for vaccine developers. The other thing we decided to focus on a bit, partly because many of us had been involved with malaria, is that better? Is challenge trials, or controlled human microbial infections, CHMI, sometimes called CHIM studies. These have been extraordinarily useful in malaria for decades now, but you can also infect people ethically and with regulatory approval for flu, for TB if you're challenging with BCG, that's done by Helen McShane's group at the Jenner Institute, typhoid and paratyphoid by my colleague Andrew Pollard. COVID-19, there was only one country in the world that used challenge studies during the pandemic for COVID. That's the UK, and again, Helen was involved in that at Oxford. And of course, pneumococcal disease challenge. And that's not a complete list. There are about 20 pathogens where you can challenge people safely. See if that's better. We work with many partners. This is a list of some of our major partners overseas. Actually, many of them in Africa, but all in sub-Saharan Africa, and many in Asia as well that are not on this list. So we're very pleased to collaborate, and of course, for many diseases, we have to collaborate or we can't test our vaccines. What I would emphasize is that to play this game, you really need some important facilities, whether they're core facilities to help you make particular types of vaccine, or a manufacturing facility, or whatever. So let me just take you through some of the facilities we're lucky to have in Oxford at the Jenner Institute. The viral vector core facility is probably been the most widely used. It says over 450 viral vectors. I think that's now over 500 have been made, and all made fairly rapidly. We have an amazing small team that can put your antigen as DNA into the DNA virus that is an adenovirus and make a vaccine. So when this was done in weeks at the start of the COVID pandemic, the press were suitably impressed, and we were pleased they were impressed, but we had made about 500 other viral vectors in a similar way beforehand. This is a pretty standard approach. Now, you're lucky to have heard from Cesar Lopez Camacho about our new mRNA core facility. It's still gearing up. We think it's going to be important. We really think mRNA vaccines have a future. I don't think they're the solution to everything in vaccines. They induce very high titer antibodies very quickly, but those antibodies come down pretty quickly as well, faster than with some other vaccine technologies. And they're okay at inducing T cells, but not very good at inducing CD8 T cells. A viral vector will do that for you much better if you want to do that. Protein production expertise, extremely important. Our co-chair, Ahmed, is one of the champions of producing not just proteins, but particularly virus-like particles or nanoparticles, and that's been the key to the success of our malaria program that I'll come to in a moment. It's not the only VLP. We have lots, as shown here on the next line, Q-beta, which you've heard about a little already from Adel, AP205, cucumber mosaic virus, and so on. We do bank adjuvants. You can do this. You can buy them. You can borrow them. You can even make them. They're really useful to have because too many groups just pick an adjuvant and for the next 20 years use just that adjuvant. There are good adjuvants and not so good adjuvants, and we have a pretty good idea of which the best ones are at the moment. And the key word, by the way, is saponin. They are the best adjuvants, in my view. And of course, you need to know a little bit of immunology. Otherwise, you can't really do vaccine development. We're very lucky in Oxford. We have extraordinary strengths in different departments. We have our own core groups working with vaccine designers to measure immune responses, particularly in clinical trials. And Lisa Stockdale, for example, is here a key member of the malaria R21 team. More unusually, we have somewhere to manufacture vaccines. As you probably know, once you make your vaccine and want to take it into the clinic, it's going to cost you the best part of $2 million these days in most places to have it made. And that's just one batch to get into a clinical trial, and that's a small batch. So this is an expensive game once you go into clinical development. We're lucky to have this very small manufacturing facility at our university, which the university owns, and we've been making vaccines there since 2005, less expensively than $2 million ago. We work closely with industry. You have to. We're never, as a university, going to license a vaccine for market supply. That's not what we do. We won't sell vaccines, but we do partner with small companies, even with foundations and charities, and occasionally with very large companies, as I'll come to. And this is just a list that I won't read through of partners we've worked with fairly recently. Some of our favorite vaccines are viral vectors. I'll come to them in the part of the talk about COVID-19. We've been using these for a very long time. MVA, modified vaccinia Ankara, was our original viral vector. It's a strain of the old smallpox vaccine that Jenner used. A very useful boosting vector. But as a single dose, single shot vector, adenovirus really wins out in terms of inducing both CD8 T cells and inducing reasonable, not spectacularly high levels of antibodies. And you can see some of the programs, including the prostate cancer program, we've used these viral vectors for. We prefer to use adenoviruses that don't infect humans, because you have antibodies to the human adenoviruses you've been infected by. So many years ago, back in the mid-noughties, if you like, we worked with a company called Kairos to develop chimpanzee adenovirus vectors, did the first clinical trial with them, and the big advantage of them is we don't have much pre-existing immunity to them because we're not infected, by and large, with chimpanzee vectors. This slide just shows you there are lots of simian adenoviruses around that we like to use to make vaccines of various types. These are some pictures or cartoons of the structure of virus-like particles. I draw your attention to the hepatitis B surface antigen, VLP. I hope you've had that vaccine. If not, you should go and get it. What we use it for is as a platform to add other antigens to put them on the outside of this VLP, and to induce higher titer antibody responses by making a nanoparticle with a protein vaccine than using the protein vaccine on its own. We also add adjuvants, so when you get the VLP and the adjuvant for your protein, you will maximize immunogenicity. Very briefly, a word again about outbreak pathogens. We did nothing in this area till 2014. It's become very important. The platform of adenoviruses is really useful for making these, as we'll see in a moment in COVID, and we've made essentially all of these, and very few are licensed. They just need to be stockpiles to be available for use in an outbreak. Okay, I'm going to leave the introduction there and come on to the first two examples of, well, actually licensed vaccines that we've contributed to developing in the last few years. And the first is a story some of you will know well about making a vaccine that was licensed here in Egypt for COVID-19. It's a viral vectored vaccine, and we were pleased, we didn't do this analysis, to see that at the end of 2021, over a year after vaccines had first been provided, an independent analysis by Imperial College London identified that this was the vaccine that saved most lives in the worst year of the pandemic, which of course was 2021. And that number was really pretty staggering. It was about 6.2 million lives. And even if you do malaria, even if you do HIV and make a vaccine, I'm not sure in your lifetime you're going to be able to make a vaccine with that impact. It really was an extraordinary opportunity. After all, only 0.6 million people a year die from malaria. It would take you a very long time to have that impact. And of course, we didn't do this alone. We did it with fantastic partners, particularly AstraZeneca and the Serum Institute of India. So I'll tell you a little bit about this story. So there's the cartoon that's been published. Pfizer came slightly second, non-significantly different, I think a scientist would say. They saved about 6 million lives as well. The Chinese coronavirus vaccine, well back at 2 million, Moderna under 2 million, and then the other viral vectors. And interestingly, even though mRNA got a lot of press during the pandemic, viral vectors actually saved more lives if you go and add up the 4 viral vectors on that slide. So how do these viral vectors work? Well, the first thing you need to understand is that an adenovirus has about 35 kilobases of DNA, lots and lots of genes. Some of that is redundant. You can take it out. You stop the virus replicating. So these are non-replication competent viruses. And in the E1, E3 gene deletion region, you put in instead whatever antigen you want to express, whether it's from cancer or from COVID or from malaria or whatever your favorite disease target is. And this wasn't a new idea. Adenoviruses first went into the clinic in around about 1990, I think the very end of the 1980s, as the well-known Ad5 vector. That's a virus that you probably have been infected with. And it was soon found that they induce strong B-cell and T-cell responses after a single vaccination. And in fact, they're still probably today the best way of getting a strong CD8 T-cell response, which you might want for whatever disease you're trying to target. But since we did the first chimpanzee adenovirus vector immunization back in 2007, at least 16 different chimpanzee adenoviruses have gone into clinical development, not just by us, even though Oxford has done most of these, also GSK, Pfizer, AstraZeneca, of course, now, and Vaccitech, a spin-off company that I co-founded. Been given to thousands of subjects, and rather unusually, the great majority of people who'd had a chimpanzee adenovirus vectored vaccine at the start of the outbreak in 2020 of COVID-19 were living in Africa, because this vector had been developed initially for Ebola and for malaria, as well as for hepatitis C by Ellie Barnes, who you've heard from. So a lot of safety data on this vector platform, unlike mRNA, where there was essentially no safety data in humans before the use for COVID-19. And that added reassurance. And certainly our regulators were happy that we were using that technology because at the MHRA regulator in London, they'd seen already 10 different chimpanzee adenoviruses. This is one of them, one that's pretty relevant to COVID-19. This is against a virus called, or a disease called MERS. It's the Middle East Respiratory Syndrome coronavirus. And Sarah, working with Pedro, who was leading the clinical trial, had generated these data showing that with the MERS coronavirus homologous protein to the spike protein, you could get good antibodies, shown in the top right-hand corner here, if I can point to that, maybe not. And in the bottom right-hand corner, very good T cells. And it didn't matter too much exactly what dose of that vaccine was being given. Now, we had no efficacy data in humans with the MERS vaccine, but have now in camels, and that looked promising. So we were very well placed in January of 2020 to make a similar vaccine against COVID-19. This was done by Sarah Gilbert in collaboration with the company Vaccitech, and looked for an industrial partner, and that was not easy. We needed an industrial partner because we were faced with the huge challenge of trying to manufacture enough vaccine, not for a few hundred or a few thousand or a few million people, but for, well, a few billion people, as you've seen from the earlier slide. This was our first vaccinee. She was a postdoc in the zoology department. I was woken up at 4 in the morning by a colleague from India saying, "What the hell happened?" And he had read this social media post shown here saying that she had died immediately after vaccination as the first volunteer. And the other vaccinees were struggling for their lives, apparently. And this is complete rubbish, of course. And rather helpfully, she herself went on social media, put up the morning's newspaper in front of her and said she was very much alive. But this was just a foretaste of what we were going to get with anti-vaxxers because of the publicity about the vaccine. We had guys up on the roof of our institute, and it wouldn't come down. And the university's official policy is to leave them there, and they're kind of right. Eventually, they did come down. But it was high profile, shall we say. The vaccine was immunogenic. It gave good neutralizing antibodies, as we had hoped, at different doses. I won't go through these data, but you can see that after a single dose, 28 days, you're getting good titers. And we then had to show the vaccine was safe and effective, not just in the UK population, where we were used to working, but with partners in Brazil, 11,000 subjects there, 12,000 in the UK, 2,000 in South Africa, 32,000 in the United States in a trial led by AstraZeneca, and then a small trial in India, whereby then most of the vaccine was being made. So by the end of the year, we had 60,000 subjects safely vaccinated. What did the efficacy look like? Well, it depends on exactly what you measure. The common endpoint was clinical disease, and our readout on that was 70% on average, 90% if you had a long interval between 2 doses, and that turned out to be very relevant for deployment, 8 to 12 weeks being ideal. And in the United States, where the interval was shorter in that trial design, the efficacy was about 85% in older adults. And by and large, the more severe the endpoint you're looking at, the higher your point estimate of efficacy turned out to be. That was doing a classical clinical trial across continents and 60,000 people. What I'd really like to do if we were doing this again is this type of study. This was done by a smart epidemiologist in Scotland who got all the hospital records for Scotland over the month of January 2021 onto his laptop. It told you when people were admitted to a hospital in Scotland. He also got the vaccination records, and very rapidly he calculated the efficacy of the vaccine in preventing people having to go to hospital with COVID-19 in Scotland. And what we really envied was his sample size. There are only about 5 million people in Scotland. 4.5 million were on his laptop. 500,000 had had a dose of either the Pfizer or the AstraZeneca vaccine, as our vaccine was now being called, followed up for 42 days, and the efficacy was pretty clear. It's on that slide, 82, 85% for each of these vaccines with a single dose. A super cost-effective as well as extremely rapid way of monitoring the real-world effectiveness of a vaccine given in programmatic deployment in one country, and a subsequent study in England and elsewhere confirmed that sort of efficacy. But then let me briefly come to what I see is probably the biggest problem, not just for this pandemic, for COVID, that's just finished, hopefully, but for the next pandemic. Because we didn't have manufacturing places in the world to make vaccine for everyone. We certainly didn't have places to make mRNA vaccines, which hardly anybody had ever made for anything. Most of the world had never heard of chimpanzee adenovirus vectored vaccines. Those were the two major types of vaccine that saved most lives, and places hadn't manufactured them. And even worse, there weren't enough places around, even if they all started manufacturing vaccines that they'd never learned how to manufacture before. So let me spell out this problem, because the point I'm making is this problem hasn't gone away. We haven't solved it. If there's a new pandemic tomorrow, we can't manufacture enough vaccine. Now, the first bizarre thing you have to learn about the vaccine market in the world is that 80% of sales, of vaccine sales by revenue, are in rich countries, highly developed countries, mainly in Europe and in North America. Most of that money goes to 5 big pharma companies. It's now going to 4 because Janssen have just pulled out of vaccines. That's Johnson & Johnson. There are about 8 billion of us now, 7.8 when I made this slide a year or so ago. And the largest selling vaccine ever prior to 2020 was almost 500 million doses, 400 and something million. If there are 8 billion of us and we need 2 doses each, you're going to need 16 billion doses. If you vaccinate everyone, if you leave the children out, it'll be a bit less. But who's going to manufacture that? So the other reality is that the great majority of vaccines made in the world today and every day are made in Asia, in countries like Korea and China and India. They're not made in South America or North America, small amount in Europe, very little in Africa. And Africa was most left behind in terms of vaccine supply, not least because of the lack of vaccine manufacturing capacity. And never before, as I said, had a tenth of the required scale been implemented. And we knew that as soon as there was a licensed vaccine, there would be an absolute clamor for the vaccine tomorrow, please, not the day after. And of course there was. And unfortunately, the CEO of AstraZeneca, with the best intentions, said that he would manufacture 2 billion doses in 2021. That was true. What he didn't mean is he'd have a quarter of that in the first quarter of the year. He meant it was backloaded towards the end. So there wasn't enough vaccine in the first quarter, even though this is one of the first vaccines available. So what do we try to do? Sandy Douglas, who was leading on the manufacturing process development at Oxford, identified some partners, companies who were very happy to help. 3 locations in the UK, some in India, Netherlands, China, and India. And in India, this was happily the world's largest vaccine manufacturer, the Serum Institute of India. AstraZeneca then came in a month later, in April of 2020, after some encouragement, and they found over 20 manufacturing sites right around the world. So we thought we were pretty well placed. The problem then, by the way, is if you have 25 manufacturing sites, you have to convince every regulator that the stuff made in Korea is the same as the stuff made in Brazil, the same as what you made in England, Netherlands, and everywhere else. You can do that, but boy, it's a lot of work. But that was what Sandy was doing for the first several months of the year, and famously bringing in the Serum Institute of India, the world's largest manufacturer. Why were they working with a bunch of academics from England who had never made a vaccine? I'll come back to that. It's the last part of the talk. Here's the map of where we were manufacturing, pretty well everywhere except in Africa. And we did look at African manufacturers. We looked here in Egypt. There wasn't somewhere we could convince AstraZeneca was going to be quick enough. But the good news was we did manufacture enough that year to distribute vaccines to 184 countries with a lot of effort. 1.3 billion doses made by the end of August, 2.3 billion by early 2022. And the key to that was one of those 25 manufacturing facilities in India who made 1.8 billion doses. They actually made 3 billion in total, then ran out of buyers. This was deliberately the lowest cost vaccine at $3 a dose, not because AstraZeneca demanded that, but the head of our university, take note, demanded it and said, whoever you partner with, the vaccine should be at cost or close to that. And that's why it was a few dollars a dose, not a few tens of dollars like the mRNA vaccines. And this turned out to be the biggest vaccine distributed by the very impressive COVAX program put together by WHO, UNICEF, Gavi, and CEPI. This is just a shot of the single room in Asia. I've been whinging that we don't have places to manufacture. One room, okay, with 6 10,000-liter bioreactors, you can see it here, and this bunch of people here who are amazing. I visited them shortly afterwards. They made 2 billion vaccine doses in a year. If we had 10 of those rooms and teams, we'd be covered. At the moment, we have 1. We could change that. And what we really want to change is this inequity. If you asked me at the end of 2021 what vaccine you had, I could tell you the answer with reasonable certainty if you told me where you lived. If you lived in North America, you would have had an mRNA vaccine. If you lived in a low-income country in Africa, you would have had almost certainly our vaccine. If you lived in China, you'd have had Sinopharm or Sinovac and so on. So there was a real geographical skew of where vaccines were going, and there wasn't good access. And worst of all, if you lived in Africa, I could have told you, you almost certainly weren't going to have a vaccine by then. And that was sub-Saharan Africa mainly. So we've got to do better than that next time. We've seen the problem. I'm going to tell you what I see as the solution. So, 2 last slides on the pandemic. Some lessons. What helped? Well, obviously, having done vaccine trials before. That makes sense. Access to experienced trial centers elsewhere. In Brazil, we're brilliant. So were South Africa, US, and so on. Regulatory support. Some regulators knew a lot about this technology that was being used. Others knew almost nothing. Luckily, they talked to each other quite a lot, which really helped. Safety data on the vaccine platform. If it was new, you had to be slower. mRNA was for that reason. And doing efficacy trials in many countries helped reassure regulators and helped us get an endpoint. We were really worried that we'd missed the pandemic in the middle of 2020 as the cases went down. Sadly, we were wrong. So what was essential? A rapid technology platform. We had one that had been developed over 10 years, access to those sites, regulatory support, funding. We got most of our money from the British government, who got some priority access as a result, and manufacturing experience with a rapid protocol I've talked about. What was difficult? I've talked about manufacturing. The world had less than 10% of the capacity it needed. That is still true. Nobody, including Tony Fauci, who was on TV a lot, knew how long it would take to make these vaccines and get them rolled out. He was too pessimistic. It was less than a year. Different regulators wanting different things is a nightmare if you're only doing one trial for global supply. And all sorts of ethical issues. The Chinese were vaccinating their military long before they tested their vaccine for efficacy. Most other countries frowned pretty severely. I'm not sure that's wrong, but I'd like people to come to a consensus outside of a pandemic as to whether, if we have a really bad pandemic and we can just make a vaccine, it's ethical to vaccinate people before there are efficacy data. Happens in the movies, could happen in real time and real life. And nationalism was a total pain. Regulators, for example, saying, do a trial in my country, we're very important, or we won't license your vaccine. If 180 countries said that, there wouldn't be very much vaccine rolled out. And the biggest countries of all with nuclear weapons only licensed their own vaccines. China licensed Chinese-made vaccines, America licensed American vaccines, Russia licensed only Russian vaccines, and I think they all thought they were right, or at least they claimed they were. So finally, a to-do list before the next pandemic: build more manufacturing capacity, agree on how we're going to distribute vaccines as a global population, not as a rich world or a poor world, sort out the ethics of vaccine use, and work on information management. You know, I'm sorry for anti-vaxxers. I'm not angry about them. I just think they need better information and education, and that can be done, and we need to start working on it. So that's the segment on COVID, thanks to, well, 300 people in Oxford and 2,000 around the world who helped with the program I've summarized in 15 minutes. Finally, a dose of malaria. We've been working on this a while. We did our first clinical trial in 1999. That was last century. We'd been working since about 93 on a vaccine candidate for malaria. The problem hasn't really got much better. There were huge numbers of deaths in 2000. In 2020, there were still huge numbers of deaths from malaria, 240,000, mostly in children and mostly in African children. Hundreds of millions of clinical episodes across 84 malaria endemic countries. Drugs and insecticides are being used well, but there's still all those deaths and malaria cases. And we're spending $4 billion a year controlling malaria without a vaccine in use. And there were, of course, more malaria deaths in Africa than COVID-19 deaths in every year of the pandemic, and, of course, continue to be every year since. This is my way of saying it's been hard to make a malaria vaccine. This is the earliest paper I've found. It was 113 years ago. Interestingly, these guys were working in North Africa designing and testing malaria vaccines. Theirs worked a little bit. Since then, 144 vaccines have gone into clinical trials, and you haven't heard of the vast majority of them because they don't work. This is a diagram I pulled out from a newspaper showing how long has been taking to make vaccines. TB was super fast, 39 years, by 1920. Malaria is the poorest boy in the class, 142 years and counting. In fact, we've just got there. There are now 2 partly licensed vaccines, and COVID, of course, was the quickest in less than a year. So malaria has been difficult. Why? You need very high antibody levels to protect, probably 10 to 100 times higher than for most other diseases, of the order of hundreds of micrograms per mil. And I spent many years trying to get T cells to work against the liver stage of malaria. Some success, but not enough. So cellular immunity isn't easy to induce either. The parasite is horribly complex. There are no licensed vaccines apart from these 2 new malaria ones against a parasitic disease of humans. About 100 times more genes than a coronavirus, harder to pick the right antigen. Different stages of the life cycle have different antigens expressed. There's antigenic variation and antigenic polymorphism. And the parasite has been co-evolving with us for about 30 million years. Now, I know we weren't human 30 million years ago, but we were hominoids and there were malaria parasites infecting us. It's a complex life cycle that you will have seen in the textbooks. We really focus mainly now on one stage, which is the sporozoite stage, number 1 on this diagram. The mosquito injects sporozoites into your skin, only about 10 or 20. They go rapidly to your liver and start liver stage and then blood stage infection and disease, and if you're unlucky, malarial death. If you have enough antibodies against the circumsporozoite protein on the sporozoite, you can be protected against malaria. There are 2 vaccines that can do this. A very old one, RTSS, that was invented in 1987 by the SmithKline Beecham Company, now GSK. And that, helpfully, that antigen has a repeat in the center. Just 4 amino acids, NAMP, repeated 20 or 30 times, highly conserved, and a monoclonal antibody against that repeat protects humans against malaria. So why haven't we got a vaccine? Well, we sort of have, but it's taken a while. The real point is you need really high titers of that monoclonal or those antibodies to protect against malaria. With RTSS, there's a mass of hepatitis B vaccine in the malaria vaccine. In fact, 90% of the vaccine is hepatitis B surface antigen, and that was required back in the 1980s to allow the antigen to assemble into a nanoparticle. So let's have a look in cartoon form at what they look like. So on the near side, near to me is RTS-S, as GSK's vaccine is called. It has little islands of the malaria antigen on the surface of the nanoparticle. The nanoparticle is about 22 nanometers. The great majority of it's hepatitis B, and that was needed to make the thing assemble into a nanoparticle. In 2010, Kath Collins, doing her DPhil thesis in the Jenner Institute, was able to get the CSP fused to hepatitis B to assemble into a nanoparticle. Ahmed is a world expert at doing that now. He's made lots of R21s and improved variants of R21. And this is relatively easily made in a yeast. One of the tricks we used was to move to a methylotropic yeast called Pichia or Hansenula to make the expression easier. And that is what the R21 vaccine is produced in today. And I'll very briefly show you the 4 clinical trials that have supported the licensure of this vaccine in 2 countries in West Africa. VAC-72 was a challenge trial where people are given mosquito bites to see if they're protected against malaria. And we were able to show efficacy that we'd never seen before of up to 82% with this vaccine in human challenge trials in volunteers in the UK and recently in challenge studies in Kenya, led by Melissa Kapulu. We went on to an African trial, age de-escalating in Kenya, into children for the first time, and then into infants, 92 subjects in total, and the safety was fine. The immune responses were even higher than we had seen in the UK. It's just an example of our friendly Anopheles mosquitoes, the most lethal animal on the planet that bite you and give you malaria. This is what we do to our volunteers who are rapidly treated if they have any parasites in their blood. And we then moved on to our first field efficacy trial in African children with this vaccine. This is Burkina Faso. This is in the middle of the cartoon Nanoro, our field trial site led by Halidou Tinto. Busy clinics as ever, very seasonal malaria. And this was the trial design. 2 different doses of the adjuvant, but a low dose of the antigen, 5 micrograms, in 150 children in group 1. Group 2 had the larger antigen dose, 150 children again, and then the control children had a rabies vaccine. We started back in 2019 and got our first exciting results in January of 2020. And our commercial partner, the Serum Institute of India, I think it's fair to say, were thrilled. So thrilled that they started looking at a COVID vaccine that I've already mentioned. The efficacy in that trial we saw then was about 77%. That's unprecedented in the malaria field. Most children prevented from getting clinical disease. And if you compare that with the RTS-S vaccine, which was ahead of us in terms of trying to get licensure, their efficacy is lower in that site. But we did one thing differently. We gave the vaccine just before the peak of the malaria season. That very likely contributes to that higher efficacy. So to see how well our efficacy lasts, and it doesn't last very well with RTS-S, we've followed now for 3.5 years. At the end of 42 months, the efficacy is still over 65%, which is really very encouraging, as shown here. There's been a lot of immunological analysis of what the basis for this is, and in summary, it looks like higher antibody levels that last longer, as you might have guessed if you hadn't got the data. And we now do have data supporting that view over 3 years of follow-up. The phase 3 trial, the licensure trial, is now submitted to regulators in many countries in Africa and at WHO. 4,800 children from 5 to 36 months of age. 4 countries in Africa, 5 different sites, shown here on the cartoon. And we divided them into seasonal sites and not so seasonal transmission sites. There's not much difference between them. And as you can see here, the efficacy at one set of sites is 75%, and at the standard sites, 67% over a year. Those numbers are not statistically significantly different. The average is over 70%. More interestingly, if you just look at the younger children, 5 to 17 months of age, when we're going to vaccinate with this malaria vaccine, the efficacy is up around 80%, both at the seasonal sites and the standard sites, higher efficacy than in the older children. Against severe malaria, very briefly, it looks similar, around about 75%. And in the beginning of the second year, from 12 to 18 months, similar efficacy again after a booster dose. The immunogenicity is higher in the younger infants. That's probably why they have higher efficacy with this vaccine, and lots of regimes work, and there's other information there I won't go into. Safety has been very reassuring, maybe a slightly higher incidence of febrile convulsions, but nothing else of any significant concern. And now, well over 5 million doses of R21 are awaiting deployment in Pune in India. The company will be able to make 200 million doses a year, which is what we need to control those 640,000 deaths a year from malaria. We see good thermostability, which is essential for a vaccine in rural Africa, and multi-dose vials are coming. So in summary, I'm not going to read through all of this because we've just 2 minutes left. We've seen very high efficacy, around about 80% in the younger children. No significant safety concerns after giving over 12,000 doses. And we were delighted last month to get regulatory approval from Ghana and Nigeria, two major countries in West Africa with Tier 3 regulatory authorities approving this vaccine. What are we going to do next? We're going to move on to adults who also get malaria, of course, whether you're a traveler or living in an endemic area, or particularly if you're pregnant. Malaria and pregnancy is a big cause of stillbirth in many sub-Saharan African countries. There are other groups interested in using this vaccine for elimination of malaria in low transmission areas. Transmission blocking vaccines against the sexual stage of the parasite are in clinical trials in Africa, including by Shumi Bishwas's group in Oxford. And we're very excited about a blood stage vaccine from my colleague Simon Draper in the biochemistry department in Oxford, who is going to combine his RH5 vaccine with our R21 vaccine in the coming months. So all looking good on the malaria front. I'm going to leave it there, take any questions if you have some, and acknowledge some of the key people in Oxford on the phase 3 trial. Their activities are shown there, including, of course, Ahmed at the preclinical stage, Serum Institute of India team leads shown there, and last but not least, the PIs of those 5 sites in Africa who did most of the work on the phase 3 trial, as shown here. Thank you very much for listening.
Thank you very much for this really great presentation and talk, and thank you for saving lives all over the world by your innovation. And I think there is a lot of comments and discussions for your very innovative presentations and trials. And one of the comments that, in spite there is no nationalism, but we want to be part of the clinical trials and to be partners with your institute. Especially, we have a very good clinical trial center, as you say today, and also the preclinical study in the Faculty of Pharmacy. Thank you very much again, and I think we have a comment from His Excellency, the advisor of the President, Professor Dr. Awad.
Sir, thank you very much for your excellent expose about vaccinology, if this term is right. I am not sure if it's right or not.
Yes.
We appreciate your contribution and the team of Oxford University, and we appreciate your presence with us today and during our Congress. Still, Africa is complaining, not complaining only from the endemic diseases in the Africa, or also from what you mentioned, or it's not your term, it is a term of WHO about the global health. Because during the era of COVID-19, which is still, we are complaining from the problem, not only in Egypt, we have a very good achievement in Egypt in managing the problem, but unfortunately, I am not sure it is unfortunately or fortunately, as I was the vice president of the group who is designed by African Union to facilitate the availability of vaccines for COVID-19 for African countries. Most probably it is the first time for African countries to have the money, but they have no vaccine. And as you mentioned, the production of the vaccine was very low, and for the first time, all the countries, they use the countries producing the vaccine, they cover their peoples with the vaccines, and they left a very small amount for African countries. And even COVAX, when they give some countries some doses of vaccine, it was very near to be expired. So that I am not convinced what we call the global health, because it's still not global. It is for some countries. And the future, we are looking for the future to have a very good achievement, either for malaria or tuberculosis or other diseases. But as you mentioned, the production will not enough, and I am afraid that also the availability and affordability for many countries, including the African countries, will be not matching. I am sure that you are not responsible for that, but maybe I hear from you.
Sure. Look, I agree with everything you've just said. The problem is what is the solution? There was an initiative by Bill Gates himself in the middle of 2020 saying he was going to build 7 manufacturing facilities around the world in anticipation of a COVID vaccine coming along. A month later, it all went quiet and has stayed quiet. Almost certainly, some advisors pointed out that if you could build a manufacturing facility for vaccines in 2 years, that would be a record. You'd then need to have the facility approved, which would probably take another year. You'd need trained staff who had done this kind of thing before. Where were you going to get those from in a pandemic? And the idea disappeared. So obviously, you've got to do something now, before the next pandemic, to have those facilities available. I'm encouraged by African governments being pretty passionate about this and putting money into looking at whether you can build manufacturing facilities in Africa. The short answer is yes, of course you can. The problem is sustainability. What are they going to do if there's no pandemic for the next 10 years? Because what really happens in the world today is that for Africa, low-priced Asian manufactured vaccines are sold in Africa at very low prices. That means if you're an African manufacturer, you're going to be outbid by a company that is supplying the biggest continent in the world with low-priced vaccines at a tiny margin, at a massive scale, and still making a profit. So there really is a business model problem. The other problem is, understandably, every country in Africa I talk to wants to have their own manufacturing facility. You don't need 54 manufacturing facilities. 5 would be really helpful. And people have to align, or countries have to align on that and see what can be done. We're trying to help a bit. We cajoled our, well, that's not fair. Serum Institute on their own were keen to help out in Africa. We were super supportive, and it was announced about 3 weeks ago that the R21 malaria vaccine will be manufactured in Africa. Didn't say that all of it would be made here, but there is a manufacturing company in Ghana and a site identified, and they're starting to build very soon. So that's a move in the right direction. There's really only Dakar, manufacturing yellow fever vaccine, a couple of small suppliers in South Africa. So there's a lot of room for investment and improvement. I would like to see significant funding going into supporting this. The European Investment Bank are supporting that Ghanaian initiative. I think this is an obvious country to build more manufacturing capacity in, but I think people need to keep up the pressure or this won't happen. It needs real international collaboration and cooperation and understanding that there's a problem now like there was in 2019. We haven't built any more manufacturing capacity yet.
May I ask His Excellency the President of the University and Professor Dr. Mahmoud to welcome to end the stage, and also the Vice President of the University, please.
Thank you very much for the wonderful talk. Thank you for coming. Yeah. I would like to wrap this very important session, and I would say you had 4 sessions over the past 2 days. So thank you very much, Professor Adrian Hill and the whole team from the Jenner Institute. I'm sorry to miss some of the wonderful talks, but I was lucky to catch up today with a little bit of the HCV, which I'm really interested in, but it was just the end. And our friend from Mexico and the Turabi, and of course, the best end was with Professor Adrian Hill. And I think when I came and visited you 1 year ago, we talked a lot about starting our collaboration. I don't know in which way, but through Ahmed Salman as a focal point for both of us. And I hope that this will continue into real steps. We'll sit together and speak into the details later on. Just about what has been mentioned by Professor Awad Tajuddin and your answer, and about the equity and the chances for different countries in the world. And the experience has showed beyond doubt that when we were in problem in the 2019-2020, and I think you alluded to this in your, Africa was left behind. And the proof is the 3.5% maximum, the amount of vaccination. And I think the Western world is not to be blamed for that. And I think it's our problem, and we have to solve it. And how do we going to solve it? We should have 1 manufacturing site, 3, 4, 5 in Africa. Again, I wouldn't go into this political issue. I would say, frankly speaking, as academics, we should search always for the know-how and how to make it what we call the domiciliation of the know-how, and then the perfection. Because, as you said, what if we invest a lot of money in a manufacturing site in Egypt or in South Africa or in Ghana, and then we'll have no pandemic for the coming 20 years? But you're not only speaking about immunization against, but you alluded a lot about therapeutic vaccines. So if we go into real science here, and we have real scientists, as you have in your part of the world, in this very important science for prevention, for treatment, for ongoing, for viral, for bacterial, for the One Health, including the livestock, there is a lot to do. So whether we have a pandemic, I hope that we'll not have any pandemic in the coming 5-10 years, but whether we have a pandemic or not, the platform should be ready anyway. Whether 1, 2, 3, or 4, this is for us to decide, Dr. Awad, and it will be a very high-level decision, but it's our responsibility to make sure that our youngs are aligned with what you're doing in a way, and to keep it ongoing and to build a brick over brick until we really have, in like Masri, for example, or other institutions, research institution in Egypt, capable of handling this kind of information, handling this kind of science, going forward and collaborating with a renowned institute like the Jenner Institute all the time, not only a single visit like what we did last year or like this year. And said that, I would invite you, if you accept, and of course you need some negotiation back in Oxford, that you consider this meeting as the spring meeting for the Jenner Institute in Egypt from now on, for all of you. So this will be an open invitation. You know, the IMF and the economic institution have got a spring meeting. So let's have a scientific one. So this is a decision I made as a president. We are totally committed to the General Institute for our annual Congress to come. I don't know the number, it will be up to you to decide, but it will be an open invitation. But between the meeting and the meeting, there we have a 12-month to work very hard on both sides and work together here and there. Joint research, joint education, joint capacity building. Again, I'll leave this to the expert to decide, but we are be more than willing to support the idea if you accept it and if Oxford accepted and the General Institute accepted. Thank you very much, Professor Hill, and thank all the speakers from General Institute, and thank you all. Thank you, Ahmed, for what you've done.
I'll just love to thank everyone, of course, starting from Ain Shams University giving the invitation, or the high-profile attendance, Dr. Al-Mitini, Dr. Professor Adrian Serhel, and of course, Dr. Ayman Saleh for his support and actually very hard engagements to organize everything. Dr. Al-Ghazali, of course, Your Highness, Dr. Muhammad Awad, and everyone attended the sessions. So that's really a great pleasure to build this link for the future, and definitely we are really enjoying being here. I think everyone expressed that so far, at least. And yeah, thank you so much, really, for your support to make this happen. Thank you.
Yeah, just to say on behalf of the university, and I don't represent the university or can't speak for it, we very much enjoyed being here. We can see great potential in doing things in one of the nearer countries in Africa to those than we've traditionally worked in, which would be wonderful. And thank you very much for your hospitality and your initiative in taking, inviting us to this excellent symposium. Look forward to more. Thank you.